Good morning, everyone, and welcome. It's really an absolute pleasure to welcome everybody here today. I'd also like to welcome our virtual attendees. We have a large number of people who've joined us online, and a special thank you to everyone who's come today to the Hayward campus of the FDA. And a huge thanks to the FDA for allowing us to host and co-host this meeting, which we hold once a year, and is really just the tip of the iceberg for the forum and the work that goes on here. We'll be hearing more about the forum and the current activities in a moment from Kristen. My name is Isabel Lusada, and I am the CEO and founder of the Amyloidosis Research Consortium, or ARC as it's fondly known. And ARC was actually founded 10 years ago, coming up for, and 5 years ago, we created the Amyloidosis Forum. As a patient, I believe it's always important to center what we do on the experiences and needs of patients living every day with amyloidosis. While over the past 10 years, we've seen significant advances in treatment landscape, and particularly in ATTR. However, these treatments have at best slowed progression, and we still have a long way to go to stop this being a devastating diagnosis where we can arrest the disease and reverse the symptoms that so significantly impact patients' everyday lives to allow them to live a full life. Thanks to the concerted efforts, we are seeing patients being diagnosed earlier. And combining that with the fact that we can no longer do placebo-controlled trials and need to have comparator arms, the field has got so much more complex as we think about developing these next generation of treatments. So the work that we do and the challenges ahead, it's really critical that in order for us to get there, that we meet these challenges with collaboration, commitment, and vision. And we're incredibly grateful to the FDA, who invited us, having worked with us joined a number of meetings and discussions, and realizing how challenging these rare diseases were. And we were invited 5 years ago to form the Public-Private Partnership, which is a collaboration between an organization that is a for-profit and non-profit, and includes at least one of each organization. We bring these together to convene and also create a structure for which to do the work. The goal of the forum is very specific. It's really bringing together the brightest minds in the field to advance rigorous science and to bridge the gaps in drug discovery and the development for both AL and ATTR. We are really fortunate, and this is one of the great advantages of the Zoom revolution, is that this is an international initiative. We are very fortunate to have regulators who've joined from Europe, Japan, and UK, along with the USA, as well as research advocacy organizations and experts around the world. We have had 5 forum meetings over the years to date, focusing on the different topics, which was initially we started with a focus on AL amyloidosis, and we did an overview of AL. We then moved on to novel endpoints and analysis, and imaging and amyloidosis. And last year, we brought in ATTR with a meeting focused on the evolving ATTR landscape. I want to take a moment to acknowledge our remarkable steering committee. The Amyloidosis Forum takes a huge amount of time and commitment, and I'm incredibly grateful to the expertise that's been shared by our steering committee that really guides everything we do. Dr. Adigan is our liaison at FDA, and unfortunately, she is unable to join us today, but will be joining us remotely. And the rest of the team, actually, Dr. Dunman was our first liaison who was in the FDA at the time and really drove the structure And he's also joining us remotely and has remained as a steering committee member. And Dr. Dispenzieri, Dorbala, Dr. Maurer, and Dr. Sankarawala have been really instrumental as leaders in the field of amyloidosis and shaping what we do. I'm also grateful to James Signorovich, who is joining us online as well, and Sarah Kent-Smith. The agenda today, which you all should have, and if you are remote, you will be able to, we're sharing a link just in case you don't have it, to be able to access that. We have focusing on 4 sessions, one on imaging standardization, one on prognostic factors, one on endpoint development, and one on federated analytics. We will, Kristen will explain in more detail about how the current forum and working groups are structured and the work that has gone into that and what phase this meeting is as part of that development. Just a couple of housekeeping. We will have, with each section, there will be time for question and answers. If you are asking a question in the room, please go up to the standing mic. Please say who you are. There is a button on the microphone, and so you have to turn it on when it is your turn to speak. So just do that. There is a Q&A box, and somebody will be moderating questions within the room. So if you're remote, you can also add your questions. We will have 2 10-minute breaks, and there'll be an hour's stop for lunch at 12:10 to 1:10. And lastly, the Wi-Fi code, which everybody always wants to know, is the FDA public, and the passcode is public access. I'd like to thank our supporters who are partners with us in not just in supporting the core work that we do, but also really important that as we think about the forum, that we bring together all the stakeholders, and we see industry as a vital partner. So we'd like to thank them for their participation and members from pharma who've joined us today. And with that, I'd love to hand over to Kristen Sue, who's ARC's Executive Director of Research and has been leading the forum. Thank you.
Good morning, everyone. For those of you who know me, you probably know by now that slides are not my strong point. As I was prepping for this meeting and short on time, I decided to do what I think a lot of people do today and turn to ChatGPT to help me visually summarize what we've been going through for the past few months. I prompted AI with to give me a simple flow chart, summarize what many of you have seen me already describe ad nauseum, and this is what I got. I won't lie and say that I personally haven't felt as confused and chaotic as this does, but it was slightly alarming to see it on a slide. I figured I'd keep going, asking it to simplify it, read it left to right, get rid of the cartoons, and this was the result. And so if anybody wants to volunteer to be this person leading everything, please, by all means. And so we're back to smart charts. And so I wanted to take a few minutes to just kind of give everybody context as to where we have come from over the past few months and ultimately where we're hoping to go moving forward. As Isabel mentioned, we've had a number of different discussions within the forum really targeted at identifying areas that are prime for collaboration in drug development. And so drawing from these different discussions and the key issues highlighted in these prior meetings and discussions, it was about a year ago that we began identifying where are the areas that we can collaboratively approach within FDA with clinical experts, industry scientists, that will be most helpful for clinical phase drug development with broad benefits across treatment mechanisms and with the ultimate goal of establishing forward-facing cross-stakeholder working groups that will cover both AL and TTR amyloidosis. And so we started with a list of 7 different candidate research priorities, each with a stated goal, rationale for why we felt like it would be important to tackle within the forum, and different approaches that could be taken to address them. These were concepts that we discussed within the Executive Steering Committee, refined, as well as started jotting down initial ideas as to who might be well-positioned to act on some of these and potentially partner with us on these different initiatives. And so from there, we sought broad community feedback in a couple of different ways. First, by having conversations and panel discussions across drug developers, so really wanting to understand where are the areas that will provide short-term and long-term benefit. And then with the broader scientific community, seeking input to validate the prioritization of these different topics, as well as identifying who might be interested in participating in them and have the ability to contribute. From there, we narrowed down the list of 7 initial work streams to 4, which I'll cover shortly. Within the steering committee, identified chairs, co-chairs, who would be part of each individual steering committee, and launched the working groups formally back in May of this year. And so this right here is kind of where we are today. And so each of these working groups has, over the past few months, has been charged with identifying, prioritizing, and implementing, ultimately will be implementing different workstream activities assigned with the objectives and the goals that we laid out for their specific working group. We had provided some initial thoughts, suggestions, but we really want to draw on the expertise across these working groups to identify what is most feasible and makes the most sense. Moving forward, the next stage of this, what we'll be talking about today is you'll be hearing are these initial proposals that have come from each working group. And so the goal for today is really to get broad feedback to help optimize how these proposals and projects are developed moving forward. And so I want to encourage everybody to ask questions, share thoughts, perspectives. This is all with the goal of helping to strengthen these projects as much as possible across different stakeholder perspectives. Moving forward, we'll be looking at feasibility for each proposal, so building in operational analytical strategies and plans, assessing resources within the working groups and where additional resources might be needed, if there's additional funding that might be needed for specific projects, seeking funding for that, and identifying additional resources as needed. And so the goal for this will be in the January timeframe, January 2025, that we would actually, across all working groups, decide which ones make sense to move forward initially, and which ones might need to be tackled either in other venues or potentially just at a later time point if there's additional resources that we need to procure in order to facilitate them. And so, as I mentioned earlier, we got broad feedback from the scientific community as to which of the work streams we wanted to, or made most sense to move forward. And so we did this in 2 different ways, one-on-one discussion, focus groups with industry, as well as with a survey that went out to the scientific community. And so this survey went out to a few hundred, I think it was close to 200 different amyloidosis experts, regulatory representatives, industry members, scientists, clinicians, really with the goal of getting their feedback on which work streams to move forward. And so the results I'm showing today are just one aspect of it, but we asked individuals to assess each of the 7 work streams based off of which would provide benefit for patients in the near and long term, whether they were likely to result in valuable publications, whether they provide additivity to the current research and collaboration landscape, whether there's clear value in working on these within the forum, whether they're achievable, the stepwise goals, whether they're aligned with individual and organizational priorities, and then lastly, to rank the priority of the different work streams within the forum. And so here you can see the results. These are based off of respondents who were primarily from the clinical landscape, hematologists, cardiologists, neurologists. The majority were based in the U.S., but followed by France and Italy. And so you can see there was high level of agreement across all 7 work streams, either at high priority or moderate priority, that these were of value and should be accomplished through the forum. But work streams 4 and 5, which are the 2 middle ones, focused on prognostic factors and endpoints, building the foundational endpoints for the selection, optimization, and evaluation of endpoints rose to the top, followed by work streams 2 and 5, federated analytics, and I'm missing one, sorry, standardization of imaging. And so these are the 4 work streams that working groups we've launched and will be reviewing today. Now, in terms of structure as to how this has all worked, it wasn't as chaotic as that ChatGPT slide would have suggested. Within the forum, each working group has a chair and co-chair that are either disease or technical experts, depending on the focus of the working group itself, providing leadership and giving direction to the overall working group activities. We have a steering committee made up of disease and or technical experts. Again, we want to make these as multidisciplinary as focused, specializing in both AL and TTR amyloidosis. This group provides advisory, thought leadership. They're helping to establish the working group plans, putting together the proposals that you will see today, and have been meeting monthly across the different working groups for the last few months. We're recruiting for broader members for each working group, so folks who are interested in participating, who have potentially data to contribute, willing to participate and take part in some of these initiatives. And so if this is something for those in the room who aren't participating or those online who would be interested in getting involved, this is something that we are looking for. If there are folks that you would recommend, please don't hesitate to shout. And then each working group has one ARC lead assigned, along with administrative support, medical writing support as needed. And our role is to provide operational and strategic leadership, as well as for projects that do require additional funding, identifying those opportunities and providing support. In terms of oversight across the different working groups, so we do have, as Isabel mentioned, our executive steering committee made up of academic, regulatory, and patient experts that are right now meeting quarterly, given the number of other meetings I'm asking them to participate in. We have a chair subcommittee that's comprised of each of the working group chairs, co-chairs, and ARC leads and ISA leadership as well, International Society of Amyloidosis. So these working groups, so this chair subcommittee is responsible for touching base, ensuring that we're not duplicating efforts, that we're being complementary in the approaches across the different working groups, and that there's general awareness of what each is working on. We have an industry advisory committee, which is made up of the different companies that are actively participating in the forum. Companies do have a seat on a working group, but this gives them the opportunity to kind of see everything that is happening across the different working groups and share their perspectives, again, to help us strengthen how we operationalize and focus our efforts. And so this is a group that meets quarterly. And then likewise, we have a regulatory subcommittee where members from FDA, MHRA, EMA, PMDA are invited, and again, to meet on a quarterly basis, review the activities and progress across all 4 working groups, and share their perspectives to help strengthen how we move these forward. And so this essentially depicts what I just said, and again, hopefully less confusing than ChatGPT. And so you can see each of the working groups vertically, and then we've got the horizontal governance kind of built across to make sure that there's awareness of what each group is doing. And then the last slide I'll share is just the plan for the year, so you can kind of see where we've come. You can see we launched these working groups back in May at ISA in Rochester, and are in what now is this project development phase across all 4 working groups at the bottom half of the slide. We'll be moving into feasibility and assessing operational analytical plans for the next 3 months, with the goal in January that we'll make a decision of which of these projects move forward initially, and how to do so across all 4 working groups. So with that, I'm delighted to introduce Dr. Sharmila Dharbala, who's part of the Amyloidosis Forum Executive Steering Committee and chairing the Imaging Working Group.
Thank you, Kristen. Good morning, everyone, and welcome to online participants as well. And I'm really delighted to be here to be representing and speaking to all of you about standardization of the imaging working group. So what were the goals? So as Kristen nicely outlined in her slides, much better than ChatGPT, I was really impressed with her organizational skills, particularly if you recall the last slide that she showed, which nicely laid out horizontally and vertically what each group is doing. So congratulations to Kristen, thank you. So the goals of our group were really to standardize the imaging modalities, both for AL and ATTR amyloidosis, in an effort to improve patient care, patient management, and drug development. Most of you, or at least some of you, were here at the previous meetings, and as you recall, there was a lot of discussion on imaging endpoints, and there was a lot of interest on developing these just not only as imaging endpoints, but also as prognostic markers. So the imaging group here, the standardization of imaging modalities group, works very closely with the prognostic and the endpoints committees in this particular fashion. So what was the rationale for our group was to really have imaging modalities to enable detection of amyloid deposits, not just in the heart, but in all the other organs, and to use them to evaluate comorbidities, to look at disease staging, looking at prognosis assessment with imaging, and of course, to look at treatment assessments, both for AL and for ATTR amyloidosis. But then, as you may recall from the last meeting, we had a lot of discussion on how these metrics were very important, but it was even more important to standardize these metrics in order to have any meaningful use of these metrics, particularly as endpoints for clinical trials and for prognostic markers. The entire group and committee felt it was important to standardize these measures. So that is what we have been tasked with. So what have we done? We've looked at the entire, not entire, but we've looked at the landscape. We looked at what's out there in terms of publications for imaging endpoints. We had a few phone calls and multiple email exchanges. And then finally we said, okay, so if you look at imaging, we have echocardiography, we have cardiac magnetic resonance imaging, and as well as radionuclide imaging with SPECT and PET-based radio tracers. So we said if we were to follow the timeline and have something that could be delivered, excuse me, we felt that maybe it was better to focus on one of all these imaging modalities. And because of that, the group felt echocardiography may be something that could be easy to start with, widely applicable in patients who are healthy and patients who are very sick, even in critical care units. With point-of-care devices, echo can reach the patient in any unit. So therefore, we said, let's focus for the initial part of this as an echo study. Now, we said, let's also try to design a multi-center study and look at variability, inter- and intra-patient variability of echocardiography. As you all know, compared to other modalities, like tomographic imaging methods like MRI or nuclear imaging, echocardiography is dependent on the sonographer who's acquiring the images to some extent, and then, of course, interpretation. So we said, could we do a simple study looking at intra- and interpatient variability? And Dr. Ruberg is going to give you a lot more details on this. And then we also discussed whether we should have a local read and interpretation of the echocardiograms with a core lab read and trying to understand if there's variability between the two. Then also discussed how this could be done as a multi-center, multi-site, multi-vendor, not just one echo equipment, but multiple echo equipments, using the same sonographer scanning twice versus two different sonographers scanning twice on the same patient. And one of the things that the group discussed at large was how do we do this in the most efficient manner, and we felt it was better to do it on the same day when the patient arrives, and more details to come on this. But of course, this is our very first application, and then once we see how this goes, we could then expand into looking at ECV, nuclear, and other endpoints as well. And partnering for other additional studies and more pragmatic studies would be the next thing to consider. So this is the whole roster of the Imaging Steering Committee members. I'm the chair of this subcommittee, and Dr. Ahmed Masri, who I believe is online, is the co-chair of this committee. And Jamie Zichterman, who's in the back of the room there, has been our ARC lead and helping us really through this entire subcommittee meetings and compiling the data together. We also have additional members in this group. They include Dr. Ashutosh Vekalekar from National Amyloid Center, probably also online today. Then we have Dr. Mariana Fontana from the National Amyloid Center in London, Dr. Rodney Falk from Brigham and Women's, Dr. Rick Ruberg from Boston University, who's here, who will be speaking to you, and Dr. Prem Soman from University of Pittsburgh, who is also online. And from industry representation, we have Spencer Guthrie from Astralis, who's here, and Ms. Lori Baylor from Pfizer, who's also joining us in the discussion here. From the regulatory side, we have Dr. Alex Hoffling, Dr. Luis Marzella, Dr. Monica Stanley, and Dr. Sue Jane Wang, who is here, I'm told, will be joining our discussion as well as soon as Dr. Ruberg finishes his talk. So it was really an amazing group, very active group, a lot of feedback and input. But today we are here to really hear from all of you on what we have prepared as a preliminary proposal before we can proceed to the next steps. So coming to the session 1, which is what we have been tasked with, Dr. Masri will be online, and I'm here, Sharmila Dorbala. Then Dr. Ruberg will make a brief presentation to set the stage to enable the discussion. And I encourage all of you who are here in person and all of you online to provide input, because this is what we are seeking from all of you this morning, so that whatever we prepare for a study, so to say, would be hopefully helpful to all stakeholders, all from physicians, patients, industry stakeholders, and from the regulators as well. So I really encourage an active discussion with all of you. And here's our agenda. We'll have a little presentation by Dr. Ruberg, and then we have plenty of time for discussion. So if this is true from the last time, I'm positive we'll have lots of active discussion, lots of questions, and that should be very interesting. So with that, I thank you all for your attention, and now really request Dr. Frederick Ruberg, Dr. Rick Ruberg, who's the Chief of Cardiology at Boston Medical Center and Boston University, to come and present. Dr. Ruberg.
Thank you very much, Dr. Dorbala, and it's really a pleasure and an honor to present on behalf of the Imaging Working Group. And I will say it's been also a pleasure to work in this working group and with our partners from industry and regulatory to really kind of come together and try to operationalize the charge of our working group. So the charge of our working group basically, as was nicely summarized by Dr. Dorbala, is to really establish the reproducibility of imaging-derived biomarkers for cardiac amyloidosis diagnosis and prognosis and progression. And this is a challenge because imaging has a lot of inherent variability, as I'm going to show you, and it's different than, say, for example, a blood test where you're making a single measure with a machine, eliminating a lot of the variability, although obviously some inherent range as well. Our mission really was to develop a study protocol of sufficient scientific rigor to establish the capacity of these markers to be used in the context that we hope to use them. And in so doing, as was mentioned by Sharmila and Kristen, we work in conjunction with the Prognostic group as well as the diagnostic group to help really understand specifically how the markers that we might identify in imaging could be utilized with a degree of precision that answers the scientific question. And as I mentioned, imaging is challenging. There's acquisition variability in terms of the sonographer in case of echo or the technologist who in case of nuclear MR. There's variation that has to do with vendors. There's technique variation, and there's interpretation variation. So there's a lot of potential sources of variation in imaging, and again, that's one of the reasons why it's so challenging, I think, to develop studies that establish the rigor of these markers. And just to reiterate that this working group was not designed to establish which markers would be selected, which markers are optimal. It was more to develop research protocols that establish the reproducibility of the markers in the disease that we're studying, in this case amyloidosis, so that they could then be, and that could inform the discussion from the other working groups about which markers then could be selected with a reasonable degree of precision. So why did we pick echocardiography? And Dr. Dorbala already alluded to this, but I'll just expound a little bit. First of all, echocardiography using ultrasound technology is globally available, and I mean globally literally. You can do echocardiography anywhere in the world. There are no safety concerns with echo. Maybe you might cause a little bit of discomfort with the probe pushing on someone's chest, but there's no way you really can hurt anyone with ultrasound. And the expense is relatively low compared to, say, MR imaging or scintigraphic techniques. or PET imaging. There are no contraindications to imaging with echo, really. And so MR is limited potentially contrast administration by renal function or implanted electrical devices or claustrophobia. There's no radiation concerns. So echo, because of its availability, its safety, and no contraindications, is an attractive modality to look at for reproducibility because it can be applied at the point of care and it can be applied in patients in extremis as well as patients who are perfectly healthy. Again, a limitation of other imaging modalities. Acquisition with echo is also short. In fact, I said 20 minutes here for a full study, but in some ways, some of the measures that I'm going to be showing you, you could actually probably acquire in 5 minutes or less. And with automated interpretation and automated acquisition techniques using artificial intelligence, you can even do faster acquisitions. And so I mentioned AI here, and we'll talk about that, I'm sure, in the discussion. Because in echo, probably AI techniques are more developed than other imaging modalities, both for acquisition to help a sonographer move a probe to acquire the imaging, right imaging plane, or in automated interpretation. And there's the capacity for miniaturization, the capacity to have probes that connect to smartphones or tablets that, again, could be done at the point of care, as I said, anywhere in the world. And that overcomes resource limitation. You don't need a big machine. You don't need a shielded room for an MR scanner, for example. So there's a lot of advantages to echo. So the protocols we're going to talk about today are really not designed to look at the AI applications, really not designed to, say, use a deep learning technique to differentiate disease from not disease or progression from not progression. And we're not talking about looking at second or third order imaging derivatives like in radiomic analysis. And I say that because I want to make sure that we're clear that we're focused on clinical variables that are accessible to a clinician and a patient that can be designed in a clinical trial broadly, not proprietary techniques that, say, for example, use a code that is only specifically available. So I've listed here some of the established, not all, but some of the established markers. And we're all familiar with these markers: wall thickness, relative wall thickness, left atrial volume on the structural side. And then functional measures are acquired from two-dimensional cine acquisitions, ejection fraction, a right ventricular measure called TAPSE, or tricuspid annular planar systolic excursion, global strain, which we're going to talk about in detail, the ratios of strain across segments, something called the myocardial contraction fraction, which uses stroke volume and wall volume, and then derived strain measures, looking at right ventricular strain or left atrial strain. These are all things that may be useful, and that's, as I said, not our committee's job to figure out, fortunately. But we are to design protocols that establish the reproducibility of these various markers, again, so that the other committees could utilize them. And then there's Doppler measures that use Doppler technology to look at things like tissue Doppler velocities, or E prime, or Doppler velocities that could measure right ventricular systolic pressure, or ratios of those things. So I want to drill a little bit down into global strain. Oops, sorry, go back one slide. And I don't know if it's going to cine or not. Okay, well, the moving picture is not moving, but that's totally fine. This is an example. This is an image of what strain is. So when we talk about strain, we're talking about cardiac deformation, just to make sure everybody understands we're on the same page here. Longitudinal strain is deformation of the heart in the long axis, and that's the red arrow up here. We also can look at circumferential strain and radial strain. Those are different strain measures. Now, the American Society of Echocardiography and the European Association of Cardiovascular Imaging has coalesced around longitudinal strain, or global longitudinal strain, as the measure that has the most reproducibility and the most clinical validity. And this is really largely drawn from the cardio-oncologic applications. Global strain is utilized, in addition to ejection fraction, as one of the principal measures in cardio-oncology to understand toxicity from chemotherapy. And it is utilized by echocardiography laboratories and reported so that cardio-oncologists and hematologists and oncologists can make decisions about whether to proceed with therapy or not. And there are a lot of advantages to strain using something called speckle tracking. Speckle tracking looks at the little image speckles that you have with echocardiography and looks at how they move towards and away from each other. And the advantages include that you basically can post-process a two-dimensional image, and it tends to be more reproducible than, oh no, it is playing. Okay, great. A little bit of motion picture there for you. This is an image showing how global strain is measured and this is the apical four-chamber view in a patient with amyloidosis. This is the apical strain, which is normal, and the basal strain, which is abnormal, and that's reflected here with an apical to basal strain ratio, which is abnormal, in this case, over 3. And so the disadvantages of echo using strain, though, are that there are image technique problems. So sometimes the contours are not well defined. Sometimes the image quality is poor, and that's going to be a limitation. Again, different than, say, a blood test where you're not going to have a limitation unless you don't have enough sample. So having good quality imaging is really important, and that needs to be built into the variability. The advantages to speckle tracking over Doppler is that there's no angle dependence. Doppler basically looks at sound waves that bounce into the image and out, and the angle of dependence is really important. You eliminate that problem with speckle tracking. So what does the literature show about reproducibility? So as I mentioned, the EACVI and the ASE put together a standardization task force, and they looked at disease patients, so patients who are not healthy. Healthy patients, obviously, you can get great reproducibility. There is no literature in amyloid, and that's what we're proposing to develop. This is a literature that actually a paper was published a number of years ago in JACC Imaging looking at 63 patients with infarct. So these patients actually had myocardial infarction, so they had abnormal hearts. I'll go back. This illustrates all the different vendors that were available. You can see there are many different vendors, and that needs to be part of the standardization protocol, as well as the different machines that are also available and the techniques. And here you can see on this bar, this is the absolute reproducibility in a test-retest of the different strain measures that are available with the different vendors. And I'll say that end-systolic strain has been arrived upon by, again, the EACVI and the ASE as the defined time when to look at strain, because you can actually look at systolic strain at different times. So looking at, say, for example, the TomTec method, which we're going to talk about at length, TomTec is a vendor that can analyze strain. There was an absolute difference of 4%. So 4% is a pretty big difference when you're looking at a reproduced total of 15%. So in these patients, with these TomTec patients, it was 15% for an absolute global strain, but the reproducibility variation was 4%. So almost 25% variance of the measure, which I think is significant and something we need to think about as we look at population measures versus patient-specific measures. So that said, our colleagues at the NAC published this paper from a couple of years ago that looked at different strain echo-derived measures, including strain, and put together a prediction model to identify the presence of cardiac amyloidosis in a patient with known systemic AL amyloidosis and the presence of cardiac amyloidosis in a patient with just increased wall thickness. So these are patients who could have hypertensive heart disease or hypertrophic cardiomyopathy or other wall-thickening diseases. And I just put here, you saw these measures on my previous slide, relative wall thickness, E over E prime, et cetera. LS is longitudinal strain, and here again you can see longitudinal strain, different cut points. I don't want you to kind of follow on the particular cut point here. That's not what I'm talking about. I'm just saying that understanding the variability in these measures, there was still the capacity to put together a scoring system with quite good predictability. Again, in this this is a multi-center retrospective cohort of patients with either systemic AL or increased wall thickness. And for prognosis, similarly, global strain has been useful in the small study that we published, a smaller study about 8 years ago and reproduced by our colleagues at NAC as well, showing that when you add global strain to patients, in this case, the 2004 Mayo Biomarker staging system, you could reclassify patients with the highest risk. Very similar data shown a number of years later with a much larger cohort by our colleagues at NAC. And in this particular case, they were also able to show prognostically that a change of 2%, so an improvement of 2% of strain, was better. And I should also mention strain, another confusing factor is it's a negative variable because it's a shortening of the heart. A lot easier to think about strain in absolute value. So, and we demonstrated that improvement in strain was associated with improvement in prognosis. Again, not the purpose of our cohort, our working group, but just to kind of show you that these data are out there in smaller single-center studies, and what we really need is a multi-center, multi-vendor study to really move it to the point of being utilized broadly. So over the next 2 slides, I'm just going to present our ideas. These are not granularized to a high degree, and this is what I'm hoping we'll work on in the discussion to follow. But as Dr. Dorbala mentioned, we're proposing an echo reproducibility study. Now, again, this is not like rocket science or a particularly super exciting study, but because we're not looking at, and I say that because we're not looking at outcomes here. That allows us to do a smaller study, however. We're looking at reproducibility, and this is going to be really important, as I said, as we understand how many patients we need to what the reproducibility is, so we can determine how to power our subsequent studies for prognosis and diagnosis. So we are talking about a multi-center study, potentially of about 3 sites or more, and a multi-vendor study. And we're proposing to limit our vendors to the 2 most prominent vendors, Philips and GE, but we can certainly talk about that. I say most prominent because most of the studies that have been published have used one of those 2 vendors so far in the amyloid space. We're talking about a same-day test-retest protocol. So that means a patient comes in, gets an echocardiogram, and then gets another echocardiogram at the same day, preferably within like maybe an hour at the acquisition of the first echo. So you're basically looking at kind of a reproducibility test without any room for variation in loading conditions or change in the patient's clinical phenotype. And this test-retest would be done by the same sonographer potentially, as well as a second sonographer. So one sonographer does a study, and then the other sonographer does a study, and you can look at the inter-sonographer variability. Now, we haven't talked in specific about subject numbers, but we're thinking on the order of 30 to 40 per vendor, and we'll have to determine the power there. We also discussed, of course, including both AL and ATTR patients, although you could argue that there may not be a difference there, but we need to demonstrate that. And we wanted to basically, kind of propose an analysis that would capture all the relevant echo variables that I showed you, so we could look at the reproducibility of each one of them. And as I said, we're not powering to outcomes, we're powering to reproducibility. As Dr. Dorbala mentioned, we would have echoes read at the local lab, so by the normal echo clinical workflow, and also at a core lab. So we understand the importance of core lab reads there. And we propose to use this semi-automated method called TomTec AutoStrain initially. We could discuss that. The advantage of the TomTec AutoStrain method is that it is semi-automated. As I mentioned, you just select the views, but it requires some modification, and it's vendor agnostic. And we also thought we would also measure blood pressure as well, so we could do some more transform measures like myocardial work, which takes into account the afterload condition. Cost. So we think we could do this probably pretty inexpensively because the first echocardiogram could be billed as a clinical study, and all we would need to basically do to support would be the second echo. It could even be done within the same billing code. It just becomes an issue of time. We wanted to focus on 2-dimensional, and I've only showed you 2-dimensional as opposed to 3-dimensional echo acquisition. We could talk about that, but 3-dimensional acquisitions are not as well validated, and they're also not as widely available and not as available at point of care. And then, as Dr. Dorbala mentioned, we could discuss partnerships with an AI vendor for automated interpretation. So we take the variability out of the interpretation side. The machine calculates all the variables, and you, in some ways, you eliminate that part of the variability. And so, and finally, so in analysis, we were talking about these inter- and inter-observer variability measures on this baseline exam. We could look at sonographer variability. We could look at vendor variability, Philips, GE, and we could look at, as I said, the core lab versus the local lab. So with that, I'd like to turn the floor back to Dr. Dorbala to have a discussion. Thank you very much for your attention.
Thank you very much, Dr. Ruberg. Really comprehensive and very, very clear summary, and he's set the stage for a great discussion here. And now we're going to have some time, about 40 minutes for discussion. And joining us here, Dr. Ahmad Masri, who's co-chair of this standardization work group, is online. And here we have with us Lori Baylor from Pfizer, Spencer Guthrie from Astralis, then Dr. Sujane Wang, who's out there and will answer from the microphone if she has any specific questions, and of course, Dr. Ruberg and I'm here. So the goal of this next 40 minutes is to discuss the protocol that we put forth. We welcome any suggestions to improve that, and we want to get input from all perspectives, from industry perspective, from academic perspective, as well as from patient feasibility, and of course, from a statistical perspective, how to make this a sound study. And eventually, we are, of course, interested in the regulatory perspective of how the regulators view this as an endpoint, if they can share some of their insights. So let me just start off with the first question, and then maybe I'll lead off the question, and then we'll talk to get questions online and from here. So Rick, you presented a very nice study design. Thank you so much. It's amazing. And AI is of great interest to all of us, and we do think it will reduce interpretive variability, particularly at a cohort level. At an individual level, I'm still not so sure. We don't have data out there, I think. But the question is, do we have any, so let me actually step back 1 second. So when I look at tomographic modalities, whether it's SPECT, PET, or MRI, there is technique variability somewhat minimized because you position the patient supine and then you push the button and the scanner does its thing. It gets the acquisition. So we minimize acquisition variability. With echocardiography, I feel like that is one of the challenges that we need to overcome is how do we make sure that the image we acquired the first time is in the same position, same location, same everything, compared to the second image to which we are comparing. So are you, Rick, aware of any AI for echo image acquisition standardization? Anybody from the audience as well can chime in.
Thank you, Sharmila. Excuse me. So I am aware that there are techniques that help guide the echo probe to a position that acquires, say a 4-chamber view or a 2-chamber view or a 3-chamber view. These are apical views from echo. I'm not sure that they standardize to the degree that you're asking for. In other words, and you very kind of astutely mentioned that when we do, for example, cardiac MR, the patient's in there's an X, Y, and a Z plane, and you know exactly where the heart is, as you put the patient back in, and you can use external fiduciaries. Maybe that would be the same thing we'd have to do in echo as well. We'd have to figure out some external fiduciary and then make sure we're in the same place. But so there is technology that can help in image acquisition, but standardization, I think, it still comes down to kind of like, well, that looks like a 4-chamber view and that looks like a 2-chamber view. But I'm not aware of any echo techniques that actually, while they assist in acquiring the views, provide the degree of standardization that you're looking for. So I think we'd have to think about, again, and maybe it would be kind of like looking at an external fiduciary and making sure that things were, a same distance and the depth was exactly the same, and all those sorts of variables that come into echo. I mean, the depth and all that, the frame rate, all the variables that are acquisition variables.
Yeah, no, thank you. I think we'll have to work on standardizing this, obviously, for a reproducibility study to ensure we minimize variability from all other aspects. Any questions, comments? Anything coming from online? Okay, Dr. Moore is coming here. Is he going to have? Does he have?
I think I'm on. Great. So, I mean, all of us have participated in clinical research studies where we utilize a core lab, and they have certain rigor. So I actually wanted to maybe ask the regulators, in the world of AI, where these images can be interpreted by a machine, and my understanding, albeit banal, is that the machine is often going to give you very little variance in the same answer each time on the same image. I wonder what regulators are going to think about, phase 3, phase 2 trials in which images are read by these machines, and how are they certified and validated, and is there a process at FDA to make sure that everything is, kosher? Many of us are a little skeptical of the black box of AI, so I don't know if anyone from FDA could comment.
Thank you, Dr. Maurer. So we have, I believe, online, I see at least Alex Hoffling and Libero Marzella. So if either one of them would like to comment.
This is Lou Marzella. Can you hear me?
Yes, now we can hear you.
So I think that one of the important challenges to getting imaging biomarkers to be useful for drug development is to be able to understand and minimize the variability factors. And certainly in the abstract, artificial intelligence does have that potential to be able to understand what the sources of variability are and to help in the precision of the measurements. So it's something that we should be looking at as a way to decrease variability.
Yeah, thank you so much, Dr. Marzella. I believe Dr. Grodin had a comment, if you would like to speak up.
Hi, everyone. Camera's not working just yet, but yeah, I was just mentioning that, while this is not AI-based standardization of imaging, there are techniques that generate an echo-based phenotype. There is commercial companies, academic individuals that, kind of generate some risk score based on imaging characteristics that are, amyloid-like, and right now they're used more for diagnosis finding, but, this is something that we would bring up in the cardiac endpoint subcommittee, but could in themselves be their own phenotype. But obviously the question then becomes, how standardizable? Sorry, that's my daughter screaming. But those are my comments.
Yeah, thank you so much, Dr. Grodin. I think this brings up an important point for our group, I think. Oh, I see Dr. Alex Hoffling there. Did you have anything to comment about?
Not much to add, just to touch on a recent email thread that we had. Our colleagues in the device center at the FDA, CDRH, they regulate software, and probably have thought about this issue of how trustworthy is AI, maybe a little bit more than the DIRM regulators on this panel, but so hopefully hoping to get them involved as we move forward with our project.
Yeah, thank you so much. One question I always had, I see many industry representatives here, many of whom have conducted studies and probably have data on echo as well. So I always wondered, even if it's an echo black box technique, is the echo image able to see change in the heart, whatever it means, for whatever it's mean, after treatment, which is not detected by GLS change, for example, or which is not detected by change in LV mass or wall thickness. I don't know if anyone has looked at it or if there's an interest in looking at a black box variation in echo measures before and after treatment for the existing data. So anybody willing to comment from the industry? No? Okay, all right. So this would be an important question, I think, because I do wonder if humans are not able to see the small amount of changes and whether AI may be better at detecting smaller changes, even if it is a black box technique to start with. We can add some reinforcement learning later on to better explain the changes. But
I can add. I think that's one of the great potentials of AI is, so again, I presented established clinical variables, but, and I alluded to the fact that I'm not talking about, say, for example, a deep learning technique that would be a black box technique where you can get attention maps, so you can know what the deep learning algorithm is looking at, but you'd need, and you'd have to figure out how you would design that. That would be, would that be a supervised, sort of model where you said, okay, these people are clinically getting worse, so is there an echo change? Or would you say, is there a change here, and can you basically, in an unsupervised manner, differentiate this from this in different groups? So I think you need a lot of images for that, and that's maybe why you asked about existing clinical trial data, and then reapplying something like that to serial existing clinical trial data, which, to my knowledge, hasn't been done yet.
okay.
Yeah, hi, John Vest from Alnylam. Could you just maybe expand a little bit on what you're talking about by black box and how that would work? I mean, it certainly across companies will now be amassing huge amounts of imaging data, and we're going to talk later about things like federated analytics. And this, I'm interested to hear a little bit more about what you're getting at with black box.
Yeah, thank you. So yes, I realized that the Federated Analytics group may be discussing this as well. So my question was primarily, could we just look at all the echocardiograms at baseline and follow-up and do a per-patient comparison, baseline to follow-up per-patient comparison, and try to see if the echocardiogram, if the AI method detects any change? It could be what they would call black box, in the sense we won't know whether it's a change in GLS or whether it's a change in wall thickness or mass or diastolic function or whatnot, but it can give us a number and say, this looks like a responder or this doesn't look like a responder, some kind of a number. And then we could refine it further. I believe, I'm not the AI expert, but I'm working with a few of them, and I'm told that we can refine it further to then ask more deeper questions of, yes, you see a change, now what does that change mean? And that's where we are. So sorry, Spencer.
Yeah, no problem. Thanks. It's a very important topic, and I think, in general, industry is very interested in how we can incorporate AI to, detect signals. So I think if there's any way to incorporate it, maybe in post-hoc analysis, the study you're doing here, because if you're able to show that there's less variability with the AI reads, then I think that will be a push for industry to look, when they look at echo longitudinally, to do an analysis using AI. So if you can detect a smaller change that's more meaningful with AI versus humans, then of course we'll want to, jump on that.
Yes, thank you.
And as you know, we're looking at that, within our own company, we're looking at that with PET, and
yes,
at least early, it does look like retest looks better.
It looks
with
better.
AI.
Yeah, that's what I would have expected.
Can you hear me? I'm not
Yes.
tall enough.
Yes.
Yeah, so first to answer this gentleman's question, and as mentioned by Dr. Hovling, our CDRH Center has cleared some cardiac-related AI tools, but we don't have them at present today. The public information about the cleared one, there are at least 2 of them, and in one recently cleared in August, it's called Cardio AI. Interestingly, when it was cleared, immediately shortly after, the public information showed that the repeatability and reproducibility with this particular AI was much, much smaller than human measurements. So it brought up a lot of hope there, but I think more information of such probably will be needed for replicating the ability to truly assess them. I'm a biomarker lead from the CEDAR for biomarker qualification program, and this also very much related to the work we do. And there, we do have an AI submission, wanting that to be considered as a biomarker. Sometimes it's an AI tool, but it's not really measure the biological activity. So we categorize them into something called iStand. It's an innovative scientific technology and innovation. So at least a couple of them are shown in the biomarker space and in the iStand space. For the biomarker, it is much more advanced now, meaning that it's not just a conceptual framework that whether this biomarker as an AI could work or not. It went from what's called the letter of intent, which means the initial conceptual framework, whether it's something doable, and it has passed that gate. It has moved on to the qualification plan, meaning things need to have a clear layout of how you're going to show this AI is going to work for you. And so maybe sometime later you will see if it's got qualified, then we will have a lot more to learn from in terms of the reproducibility and repeatability with this kind of AI tool as a biomarker.
Wow, excellent. Thank you so much. That was very comprehensive. I hope all of you could hear it, but there are, one or two tools that are already approved, and definitely they need to be validated, I think, hopefully in a federated analytics platform or whatever. Dr. Castellano.
Hi, Adam Castagno here from Bridge Bio. So just wanted to add quickly, too, that, if we are able to consistently show reproducibility with AI echometrics, especially given the plethora of data that we now have across multiple clinical trials that have utilized echocardiography and ATTR and AL, and we're able to tie that to clinically important outcomes that we've seen in the trials, it could potentially be a way to utilize, along with other biomarkers, essentially a way to shorten clinical trials in the future. Potentially, that would be a tremendous benefit, I think, across the board, especially now as trials are becoming more and more challenging since we can no longer really placebo control. So.
Yeah, that's an excellent point. Thank you so much. Actually, that's a great point. And so I think one of the challenges that we will face with this disease is the disease is not a stable disease. It's constantly progressing. So even if you look at the placebo cohorts in all these trials, the variability may be disease progression, not necessarily test variability. So how do we deal with this? So whatever study we are planning, it will be small, but we will probably consult with Dr. Wang and others to see if there's anything we can do to up the numbers to have at least some basic information on AI. But again, my personal feeling is it works better for populations, maybe not as much at an individual level, but something to wait and see. Oh, I see Dr. Fontana lifting her hand. Hi, Mariana.
Hi, and thank you very much for pulling together these meetings. I mean, it's very kind of important conversation. I just wanted to add something about variability that also Rick alluded to, in the sense that when we think about imaging, we've got 3 different levels of variability, in the sense the variability related to acquisition of the images, which is quite relevant for echo, and less for CMR. The variability related to biological variability, in the sense a patient coming twice on 2 different days in different physiological condition is going to have an intrinsic natural variability related to actually biological variability for that measurement, and then variability on the image analysis. And when we think about AI, we are going to only have an effect on the image analysis variability if we apply AI to basically analyze the images. So for echo, for example, is at present, I think, a bit uncertain what is the weight of these 3 different components of the variability of the technique. And that's why I think the study that Rick is proposing is so important, because you will be able to untangle the acquisition variability from the biological variability, from the image analysis variability, and is really only understanding the relative weight of these 3 components that you understand, for example, the effect that AI has, because if their variability is the bigger component, AI is gonna lower variability, but if their variability is actually the one that weighs less, then the role of AI there is definitely less. And on the second point, when we think about black box approaches compared to annotation-based approaches, for example, I see a black box approach way more suited for diagnosis, where you try to create a higher pretest probability population, where then you apply your test to confirm a diagnosis. However, I don't see, for example, a black box-based approach to have a big role in progression, because it's very difficult for a clinician to say, oh, this has been flagged up as progressive on a black box approach, but is not explicable, whilst an annotation-based approach, similar to what Rick was mentioning, for me, for follow-up would be definitely more suitable, because telling to a clinician, by using AI, I can tell you that the stroke volume, by increasing precision, is getting worse in this patient. Then I think clinician, being a fully explicable annotation-based approach, they will be more inclined to say, yes, this patient is progressing because the stroke volume is going worse, which is a biologically important measure. So I think black box approaches and annotation-based approaches have got different role in diagnosis versus tracking changes over time, but we'd be interested to know also what the other panelists believe.
Yeah, Absolutely. Thank you so much for discussing this comprehensively. I could not agree more. Thank you. Dr. Quarta here.
Okay. Hi, everyone. This is Cristina Quarta from Alexion AstraZeneca. So one of the, it's not a question, but more of a comment. So I think that one important impact that this effort could have in the long term, hopefully less than long, is how to mitigate the need in the clinical trials to have these extensive qualifications for sites to participate in trials. Because imaging right now is one of the rate-limiting steps for, patients to be screened and enrolled in trials. So whether it's echo or CMR or any other type of imaging, I think that is a very important aspect that we, broadly have to address.
No, excellent. Thank you so much. So that brings up, oh, sorry. Go ahead.
Sorry, I just want to add to that. It was an extremely important point, and I think some learnings from this trial that could help is, as a sponsor, you have to kind of decide what are the most important controls we need to put in. What is the rigor, and maybe this can kind of separate those things out. So is it more important to ensure the site uses the same sonographer? Is it more important to ensure they use the same software? Is it more important to, limit the number of centers? So understanding what's driving the variability and sort of picking and, as was just stated, qualifying sites. So can you give up some things in qualifying sites, or are we going to have an answer that says we need to put in even tighter controls?
Yeah.
Yeah, this is a great discussion, and I really appreciate both the points made by Spencer, Chris, and Mariana. I think the AI point, I think, is interesting to think about in two different ways, and we've kind of already touched upon. One of the black box, I think we've talked about. The problem with the black box is, as a clinician, you don't really know what, that may mean something to you or may mean nothing to you, And it also kind of gets a little bit away from our concept of multimodally assessing patients, looking at biomarkers, clinical exam, and other sorts of, how patients feel, So I think the advantage of just using a standardized quantification method, then you will know, and if we establish the validity of that, then we'll know what exactly is changing, and with a degree of precision that then can be incorporated into a kind of a more multimodal assessment, which is why I brought up the publication in my slide presentation about how it's utilized. That was all echo. There was no input from biomarkers or clinical exam or anything like that. But you could imagine that ultimately our assessment is going to be a combination of things, of which imaging is one, and our task is basically to establish its reproducibility. So I feel that some of maybe the approved or soon to be approved, if there are any modalities that basically kind of say, we can take the variability out of the interpretation side, just as Mariana said, and know stroke volume is this, with this reproducibility, GLS is this. These are the variables potentially then that can be incorporated into a larger model that might use proBNP or troponin or clinical exam or whatever, EGFR, to try to understand if a patient's getting worse or better.
Yeah, no, I fully agree. I think the black box, when I brought it up in the context of change, is if it shows no change at all, and if it confirms what we're seeing visually, there's no big change, I think that would be useful to know as well. But if it does so change, again, what I meant by that reinforcement learning is really to get more deeper into understanding what exactly the change means or does not mean. So I'm actually very pleased to see the interest from everyone, because when we started this, one of the questions that we had amongst the group is, would this study be useful at all doing EPO reproducibility? Because, the ASC people have guidelines, CVI has guidelines, should we do this? When we entered it, we said it's a simple question, but this has not been answered in a multi-site fashion, specifically for amyloidosis as a disease for which we have so many new therapies coming on board now. So that's why we felt it was important to do it, and I'm happy to see that there's a lot of engagement and interest on the discussion. So, Rick, would you agree with that?
I completely agree, and I think your point before, Sharmila, about how there is going to be natural progression in disease, it it's different than, say, myocardial infarction, where an event occurs, and then theoretically the patient's not going to have another infarct in the time between you do your two studies. So I think our disease is being a kind of a progressive disease, you really need to kind of understand like treatment and non-treatment and understand, so I think it's almost that this is the homework that needs to be done before we apply these techniques to show in the disease that is progressing, how reliable are these measures. And so how do we make sense of a 2% change in the context of a 4% variation in an individual patient versus a population of people?
Yeah, and one thing we were discussing before we came on board, just for the sake of everyone, I've been involved in some longitudinal studies in AL amyloidosis patients, and I can tell you that the patients who return, we do see improvements or some changes, but those are the survivors. Okay, the ones who did not see improvement, the ones who are worsening, are dead or too ill to come back for a cardiac MRI or a PET scan, which is what we studied. So which is why I think it becomes very, very important for a technique like echocardiography, because this can go to the patient. The patient need not come to the scanner. We can go and scan the patient wherever they are using this technique. And it's very important to recognize that this survival bias, the bias that the publications where you see that improvement indicates good prognosis, absolutely does, because these are survivors who came back for repeat imaging, who are alive, who proved themselves to be alive.
So great project and great discussion. The question I would ask, and this is more for the regulators, in these other fora that we've had, endpoints have always seemed to be needed to be functional. So I would ask, I think this is important to understand how we use our tools and what the limitations of our tools are and so on. But I would ask, to actually use it as an endpoint. Will there be buy-in? Because functional seems to be key, and echo isn't functional. So that's my question.
This is Lou Marzella. If I could try to address, I think it's an important question you're asking. And I think that this has to do with the what we would call clinical validation of the imaging technique. So this group here is being focused on the technical validation aspects, and I think the technical validation aspects are fundamental to understand the variability of the measure. But then the next question is the actual measurement fit for the purpose that it's intended to serve, in terms of not only how small a change can be detected, but what is the meaningfulness of that change. So I think that the work of this group has to go hand in hand with the other groups that are sort of focused on what would be the endpoints that this imaging test is supposed to be assessing, and what is not only the minimally detected change, but what is the minimally clinically meaningful change that can be detected. So it's a complex interaction, but it has to start with the standardization and the technical validation of the methodology before proceeding to the what we would call clinical validation.
Thank you so much, Dr. Marzella. Yeah, so I don't know if anyone online has comments. Dr. Masri, others? No. All right, so one of the other, sorry.
No, I was just going to echo back that I think even if we, in an ideal perfect world, had an AI tool to minimize all variability, let's just pretend that we're able to do that. I think that's a really great point. If we minimize the variability, it still becomes a question is what is a clinically meaningful change? What's a statistically meaningful change? But more importantly, what does that translate from a clinically meaningful change among these patients? And do you see that to be different among, like we talked about earlier, are there variations of what we see in AL patients versus ATTR patients? Maybe, maybe not, but I think that'll be important to make sure that the data supports that as well.
Yeah, excellent points. And actually coming back to what Dr. Angela Dispenseri mentioned, Functional endpoints are important, and we are, especially with ATTR amyloidosis, we are looking at very old individuals who have multiple comorbidities. So GLS or changes in GLS may not represent just amyloid, but could be high blood pressure, diabetes. Maybe they have other comorbidities that are impacting their GLS. And from a science perspective, I think we want to see what amyloid is doing or how it's changing. But from a patient perspective, I feel it's important that every patient does better. If they have comorbidities plus amyloid, we still want to see them improve. They don't have comorbidities with amyloid. So I don't know if I'm making sense, but I think I like GLS for the fact that it is representative of multiple things going on to the patient, and it may be more valuable from a patient perspective for that reason. Dr. Charu Gandotra?
I can figure out how to...
We can hear you.
You can hear me, okay. Hi, I'm Charu Gandotra from DCN. So just adding from the drug perspective, potential acceptability of one of these imaging biomarkers or other biomarkers as an endpoint to support approval. So there are two pathways. One pathway is where a biomarker is being considered as a reasonably likely biomarker, where one will still require a confirmatory trial based on clinical endpoints, field function, survive metrics. And then the higher bar, which is a validated surrogate endpoint, where we need a lot more data and assurance that that biomarker truly does translate into clinical outcomes. So what we really want to understand, if you're thinking about a biomarker to accept as a reasonably likely or a validated surrogate endpoint, is to see
Yeah, absolutely. Absolutely. I'm sorry. I think your microphone wasn't functioning towards the end, but it's a really, really great point. We have about 4 minutes. Okay, one more question. Go ahead.
Oh, great. Thanks. Donna Grogan from Protego. I applaud you on this study. I think the prospective nature of this allows you to do the test-retest variability of obtaining the test at the same time. But I'm a little bit concerned of the size of the study won't be able to answer the questions that you're trying to answer, particularly around the biological variability that was just discussed. And, ATTR and AL are both amyloid, but they're different diseases, right? And cardiac Mayo classifications, does that impact the variability of the measurements and the interpretation? And it seems like you have a wealth of information out there from all of these trials that have all these echoes. Now, they don't have repeat echoes on the same day, but you may be able to get all the other information from all of the data that's already out there. So I would just encourage you to, as was mentioned, try to get some of these other data sources to try to inform the questions you're trying to answer.
No, great points. Thank you so much. And I fully agree about the size of the study. Dr. Wang has a comment.
Yes, I was debating whether I should comment on this part, but since I'm part of the working group, I think I should.
Yes.
Yeah, so when we talk about reproducibility, repeatability, that kind of, whether we call that technical performance or analytical performance, we generally don't do it as a power calculation. Mm-hmm. So sample size-wise, I very much agree with the comment just mentioned. This might be a place where we can leverage trials that actually need to use echo, mm-hmm. and go from there to design a sub-study, very much like imaging trials when this type of reliability measurements are needed. They generally are a sub-study of a larger trial. And from there, the larger the number of sample size you can get, meaning the number of subjects you can get, the more accurate the estimate of how that reliability will be.
Yes. Thank you so much. Thank you. I think we have 1 more minute and we need to wrap up. So thank you all for the very valuable feedback. So maybe I'll do a quick show of hands. We propose to do maybe 1 or 2 vendors. How many of you agree with using just 1 or 2 vendors for the echo sub-study, or do you think we should look at maybe 4 or 5 vendors? Show of hands. 1 or 2? Sure. Okay. All right. So we will probably talk to all of you by email more, because we need sites with other echo machines. So the limitation is we're not trying to restrict 1 or 2 vendors, but if you're out there, a site using an echo with not Philips or not Siemens or not GE, then reach out to us, because we need you in this type of a study. We would like to keep it as broad as possible. All right. With that, thank you really to the panel, to all of the audience for active participation, and to Kristen for organizing this session. Thank you all. And Dr. Ruberg, do you have any comments?
No. This is a wonderful discussion. It was great to hear all the different inputs. I think kind of it epitomizes this working group, which I think is great. Thank you for your leadership.
Thank you.
All right. So we will take a break and reconvene at 10:15. So 10-minute break. Thank you. All right. So for the second session of the day, I'm delighted to turn this over to Dr. Matt Maurer, who I feel like does more for ARC than he must have time to possibly do, but I'm thankful to have him chairing the Prognostic Factors Working Group. So, Dr. Maurer.
Thanks, Kristen. I have to say there's probably nothing better than taking care of an amyloid patient, but it's great to see all colleagues here. It's a wonderful field, and it's, I think, a credit to ARC that we're kind of galvanizing and get everyone together to move the boat forward. I have the pleasure of chairing the Prognostic Factors Working Group with Ashu Welkaker. Ashu, I think you're on if you wanted to say hi. Just to everyone, albeit remotely.
Remotely.
Great. So let's see if this will click forward. Maybe not. There we go. So just broadly, our working group is obviously focused particularly on prognostic factors for drug development, but also for clinical management. I think all of us who take care of patients, their most common question to all of us clinicians is, how am I doing and how am I going to do in the future? And this is really near and dear to the patient's heart, and so it's something that we really do need to focus on. Our broad goal is to try to develop validated prognostic factors that could be then shifted over, hopefully, to the Clinical Trial Endpoint Committee that focus on kind of qualitative assessment, but endpoints that are in this arena of this rapidly changing landscape in the standards of care. And I think the rationale for this is well stated here. We want to understand prognostic factors, which, as we all know, are central to clinical trial design. We use these factors for inclusion-exclusion criteria, for risk enhancement, baseline stratification, determining trial duration, possible use of external controls, so that's hard in a rapidly changing landscape. We're interested also in trying to not only look at each individual prognostic factor, but combine them into potentially composite scores that could be utilized to help patients understand how they're doing on a particular therapy, but also in the design of clinical trials. And I think we're kind of interested in this discussion also to hear how we could learn from other organizations so that FDA or regulators deal with, and lessons learned in maybe adaptive trial design, and trying to figure out in this rapidly changing landscape how we could alter a trial with some confidence that we're still obviously determining the safety and efficacy of a particular drug or compound. I think we have emerging confidence about prognostic factors for outcomes, but those are usually outcomes focused on, unfortunately, survival and mortality, and we all would like to know things way before that particular endpoint, certainly. And so, what is the relationship to other prognostic factors to other clinical events, functional capacity, God forbid hospitalizations, and so forth. And I won't enumerate the possible approaches because I think you'll hear from my colleagues today about that. This is a fantastic working group. As I said, Ashu and myself have the privilege of co-chairing it, and the members are shown here. Most importantly to note that we have representation from the important disciplines of cardiology, neurology, nephrology, industry, regulatory, and can't thank Kristen enough for all her efforts in what is often hurting unicorns, not even cats. So good for you. Today, the session will be chaired by myself and Ashu. We have actually four speakers, sorry. We took in the tact of having Raj Chaturboki initially talk about prognostic factors in the AL realm. Joban Vashna will talk about TTR. And we kind of feel like this sets the landscape for how we all could come to some common understanding of what could be utilized. As you'll see, I think from my colleagues, there's no shortage of things that we could measure, but the question is which ones are most preeminent. Jan Griffin will give us a proposal that we've developed as a working group around cardiopulmonary exercise testing, and then we will hear from Jing Zhang about an AmyLite project proposal, and there'll be plenty of time for discussions both in between all of these talks and at the end. So without further ado, I'll turn it over to Raj Chaturboki from Columbia University.
All right. Thank you, everybody, for your attention. And on behalf of the Prognostic Factors Working Group, today I will present the prognostic factors in AL amyloidosis and progression. These are my disclosures. So first, to set the stage, we will talk about how to think about prognostic factors in AL amyloidosis. And as all of you know, the prognostic factors in AL amyloidosis can be broadly divided into clone-related prognostic factors or organ-related prognostic factors. And we will go over each of them individually as to what the data is there, especially in the setting of changing therapeutics in AL amyloidosis. The most common clone-related prognostic factors that we typically think about are percentage bone marrow plasma cells, cytogenetics, DFLC, which is the difference between the involved and the uninvolved free light chain. And we have some dynamic prognostic factors, including a stringent hematologic response, for example, a stringent DFLC response or DFLC of less than 1 milligram per deciliter, and minimal residual disease status. And an important candidate for that currently is free light chain mass spectrometry for non-invasive minimal residual disease assessment in AL amyloidosis. As far as organ-related is concerned, as we all know, the cardiac prognostic factors are the most relevant when it comes to mortality, including NT-proBNP and troponin, which are an important part of both staging systems, the Mayo 2004 and Mayo 2012 staging systems. We also have functional testing, including 6-minute walk test, imaging parameters, which we have already heard about, including global longitudinal strain, percentage extracellular volume on MRI, and also health-related quality of life. As far as renal prognostic factors are concerned, they include 24-hour urine protein and eGFR, and there is also some data on autonomic neuropathy being a bad prognostic factor in AL amyloidosis. So we will first focus on some of the clone-related prognostic factors, and then we will go on to the risk stratification and the organ-related prognostic factors. So first, looking at bone marrow plasma cell. So we have known for many years, even before the advent of the novel agents, that higher bone marrow plasma cells portends worse survival in AL amyloidosis. This was a large study from the Mayo Clinic group. As you can see, with bone marrow plasma cells, there was with a stepwise increase in percentage BMPC. There is a worsening of overall survival, with patients having a bone marrow plasma cell of more than 20% having a significantly worse overall survival on multivariable analysis. This was true in the entire cohort as well as at landmark analysis at 1 year. One thing I would like to point out here is that most of the patients here got either autologous transplantation or melphalan-based therapies. A minority got bortezomib-based therapies, and none got daratumumab-based frontline therapy. So the prognostic importance of bone marrow plasma cell could definitely evolve with changing therapeutics, and more data is needed in the current era. We looked at our data in Colombia on 100 consecutive patients treated with daraxonrasib or daratumumab-based frontline therapy. And we found that as far as time to next treatment is concerned, again, with a stepwise increase in bone marrow plasma cell, more patients needed a second-line treatment. With patients having bone marrow plasma cell of more than 15%, they had the highest need for a second-line treatment. However, our follow-up was still very short, it's about 1.5 years, and at 1.5 years follow-up, we were not able to see any difference in overall survival between these three cohorts. So the question remains whether the impact of percentage BMPCs on overall survival, it will hold up in the daratumumab era, and for that, we really need long-term follow-up in these patients. As far as cytogenetics are concerned, we have learned that gain 1q or amplification 1q, that is plus 1q, is a negative prognostic factor in the era of daratumumab-based frontline therapies. And we have seen that patients with gain 1q, they have a significantly lower rate of hematologic complete response as well as a hematologic VGPR or better. And that does translate into a lower hematologic event-free survival. But again, with a follow-up of roughly about 1.5 years median follow-up, we have not been able to see any difference in overall survival between the different cytogenetic subgroups, which also speaks to the improved survival of these patients in the daratumumab era and difficulty in seeing overall survival differences between different prognostic subgroups. For that, we really need a long follow-up. One important point I want to highlight here is that T1114, it used to be a negative prognostic factor for hematologic response as well as heme EFS and overall survival in the bortezomib era, but we don't see that in the daratumumab era anymore. Patients with T1114, they actually had similar hematologic response compared to those without T1114. Then moving on to the dynamic clone-related prognostic factors, one of the most important dynamic factors that was initially described, one of the largest studies from the UK group, was a stringent DFLC response, that is a DFLC of less than 10 milligram per liter or less than 1 milligrams per deciliter at the end of treatment. And an important point to highlight here is that even in patients who achieve a VGPR, that is a DFLC of less than 4 milligram per deciliter, the stringent DFLC response does discriminate survival, with DFLC of less than 1 milligram per deciliter, patients with DFLC of less than 1 having a significantly superior overall survival compared to those with a DFLC of 1 to 4 milligram per deciliter. So even at such low levels, DFLC was able to discriminate overall survival in these patients. More recently, we have free light chain mass spectrometry as an important candidate for non-invasive minimal residual disease assessment in AL amyloidosis. And this was, again, data from bortezomib-treated patients from the UK group, showing that only about 30% of patients in heme CR with bortezomib-based treatments were negative by free light chain mass spectrometry. And those who were negative by free light chain mass spectrometry, they had a superior overall survival compared to those who were free light chain mass spectrometry positive. If you look at the figure B, those are the patients who were all in hematologic complete remission, who we know have a good overall survival. But even among patients with heme CR, those who were heme CR and free light chain mass spectrometry negative, they had a significantly superior overall survival compared to those who were heme CR and free light chain mass spectrometry positive. So going forward, one of an important prognostic factor and potentially an important endpoint could be free light chain mass spectrometry negativity in the blood after treatment. Now, going into global risk stratification, so we have two staging systems that we use in AL amyloidosis. One is Mayo 2004 staging system, and one is Mayo 2012. This is the Mayo 2004 staging system. So as you can see here, patients are stratified based on, patients are stratified based on NT-proBNP and troponin. So it's only organ-related prognostic factors. And we have stage 1, 2, and 3, and stage 3 was further divided based on NT-proBNP of 8,500 nanogram per liter cutoff into stage 3A and 3B. And this was predominantly before the era of bortezomib-based treatments. And as you can see, patients with stage 3B, they had dismal overall survival of less than 6 months. This is the Mayo 2012 staging system, which includes DFLC in addition to NT-proBNP and troponin, and divides patients into stage 1, 2, 3, and 4, with patients with stage 4 having a median overall survival of roughly about 6 months. Last year at the ASH annual meeting, Dr. Vechalekar's group presented data on more than 1,200 patients homogeneously treated with bortezomib-based frontline therapy and looked at overall survival by both the modified Mayo 2004 staging system and overall survival by the Mayo 2012 staging system. So as you can see, in patients who are homogeneously treated with bortezomib-based therapies, the overall survival is much better. By the 2012 staging system, we saw that for those who were stage 4, they had a median overall survival of roughly about 2 years, and which is much better than the originally described approximately 6 months. Those who were Mayo 2004 stage 3B, they still continue to have a dismal overall survival. It's roughly about 1 year. So it's, those are the patients on whom we still need to do much improvement. A question often comes up that in the era of effective clone-directed therapies like bortezomib and daratumumab-based frontline therapies, does DFLC, is it still an independent prognostic factor for overall survival? And there was a paper recently published by the UK group in AmyLite on approximately 1,300 patients treated between 2010 and 2019 homogeneously with frontline bortezomib-based treatments, and they had a really long follow-up of roughly about 6 years. And they found that on multivariate analysis, the modified 2004 staging system, which is basically NT-proBNP plus troponin, and a global longitudinal strain of more than -9%, and a 6-minute walk test of less than 350 meters, all of these were independent prognostic factors for overall survival. However, DFLC of more than 18 and systolic blood pressure of less than 100, they were not. And based on this, they have proposed a new stage 3C, which is Mayo 2004 staging system, plus a GLS, sorry for the typo, it should be GLS of more than -9%. And this group, despite homogeneous bortezomib-based frontline treatment, had a median overall survival of only 4 months. This definitely needs external validation in daratumumab-treated patients, but it shows that, as we have already seen in the imaging working group, that GLS could be an important candidate for a novel prognostic factor in AL amyloidosis and incorporation into staging systems. Now, switching gears into to the organ-related prognostic factors. So, as we know, NT-proBNP kinetics is a very powerful prognostic factor in AL amyloidosis, and this is quite old data now, I think from 2012, in which Dr. Palladini's group showed that NT-proBNP kinetics as early as 3 months from treatment initiation predicts overall survival. That is, patients who had NT-proBNP response at 3 months had a significantly superior overall survival compared to stable NT-proBNP, which was again significantly superior compared to those with NT-proBNP progression. Now, switching gears a little bit into functional measures. So 6-minute walk test is the functional measure for which we probably have the most data in AL amyloidosis. And as you can see, the 6-minute walk distance, it decreases with Mayo stage. And at baseline, patients who are unable to do a 6-minute walk test at all, they have dismal overall survival as expected, versus those who have more than 350 meters, they have superior overall survival. Another important thing to note here is that 6-minute walk test is also responsive to therapeutic intervention. So the UK group also showed that patients who had a more than 33-meter improvement in 6-minute walk test, they had a superior overall survival compared to those who had less than 33-meter improvement. As far as imaging parameters are concerned, we have already talked about GLS. We also have percentage extracellular volume on cardiac MRI, which is an important prognostic factor, and also it's responsive to therapeutic intervention, as shown by Dr. Mariana Fontana's group from the UK. So as you can see here, the first figure is basically the survival function for CMR response at 6 months post-chemo, and the second is 1 year post-chemo. And again, similar to NT-proBNP, we've seen that patients who have regression of amyloid on MRI, which was defined as an ECV reduction by 5% or more, they have a superior overall survival compared to those who have stable or progression of amyloid. Quickly, the renal prognostic factors. So with the renal staging system, basically it's EGFR and 24-hour urine protein, and what that predicts is the percentage of patients who will need hemodialysis at 3 years. It does not predict survival. And patients who have an EGFR of less than 50 and proteinuria of more than 5 grams in 24 hours, those patients have almost a 60% risk of needing dialysis in 2 to 3 years. Finally, autonomic neuropathy. So autonomic neuropathy is more of a subjective measure, so I could not really find a lot of data in the era of modern therapeutic interventions. This is an old paper from the BU group in which there was data from 112 patients with AL enrolled in a clinical trial, among which about 1/4 had definite or possible autonomic neuropathy, and there was a significant decrement in overall survival in patients with autonomic neuropathy compared to those who did not have autonomic neuropathy. And they defined autonomic neuropathy based on some clinical features, for example, drop in blood pressure, along with some other symptoms. So in summary, disease-related prognostic factors in AL can be broadly divided into clone-related and organ-related prognostic factors. The key baseline or static clone-related prognostic factors are percentage bone marrow plasma cells, DFLC, and FISH cytogenetics. It's important to remember the prognostic impact of individual factors can shift with changing therapeutics. The key dynamic prognostic factors are indicators of profound hematologic response and MRD status, with free light chain mass spectrometry emerging as an attractive non-invasive MRD assessment strategy in AL. Among organ-related prognostic factors, cardiac biomarkers, including NT-proBNP and troponin, are important for predicting survival, with NT-proBNP kinetics as early as 3 months predicting overall survival. And finally, renal prognostic factors, including 24-hour urine protein and eGFR, can predict the likelihood of patients needing dialysis. With that, I would like to acknowledge our AmyLite program at Columbia, and I'm happy to take any questions. Thank you.
Thanks, Raj. I think we will have a second talk, and then we'll open it up for discussion. So I'm pleased to introduce Joban Vajdov from Johns Hopkins, who will talk to us about the landscape of prognostic factors in TTR amyloidosis.
Thank you. Good morning, everyone, and I want to thank ARC and the members of the forum for the opportunity to talk today on topic at hand, prognostic factors and disease progression in TTR. So I'd like to start first by defining the terms that we're going to be diving into, specifically disease prognosis, which literally translates into knowledge beforehand. These are factors that allow us to predict one's disease course, including the likelihood of disease progression. Disease progression, something we maybe can recognize when we see it, but is essentially escalating signs and symptoms related to one's chronic medical condition. And then putting this into context of an endpoint, another topic at hand today, these are, as defined by the FDA, objective measures that have been designed to reflect the intended effects of a therapy and the safety of a therapy. And I think these definitions matter because of what was aforementioned in terms of the goal of this collaboration. And I think in ATTR, a goal would be to define disease progression and ideally align that definition with endpoint measures in TTR. So I would be remiss to talk about prognostic factors in TTR without first highlighting the dramatic evolution in the prognostic landscape, much to the thanks of everyone in this room. And this is really due to a few key advancements, one being the ability to non-invasively diagnose TTR amyloid with the use of nuclear scintigraphy, and then, of course, the availability of disease-modifying treatment. I think this change over time is highlighted nicely in a figure that was compiled by Dr. Brett Sperry, in which he superimposed survival curves from the most recent or contemporary TTR-CM trials. And as you can see, relative to patients in the ATTRACT trial, patients in ATTRibute-CM and HELIOS-B had a markedly improved survival, and we know that these patients were, in general, less sick and diagnosed at an earlier stage of disease, as evidenced by the baseline characteristics, lower NYHA class, NT-proBNP, NAC staging, et cetera, to where the treatment arm of HELIOS-B almost approaches age-expected mortality for the disease, which is remarkable. So now diving into specific prognostic factors in ATTR, I highlight a few here and put them in different domains, including clinical, laboratory markers, and imaging markers. And by no means is this a comprehensive list, but certain clinical factors that have been shown to be prognostic include, of course, age, functional status is highly important, comorbid conditions, as well as genotype. And I think in the U.S., this mostly affects patients with V122I disease who have been shown to have a markedly worse prognosis. Laboratory markers that have been shown to be important, which I'll get into, include proBNP, troponin, renal markers of disease, and emerging neurofilament light chain. And then imaging markers, which I won't dive into too much in this talk, as there's a separate working group on this, have also been identified to be prognostic across echo, PYP, and MRIs. And to highlight prognostic factors further, this is work that was published by the NAC cohort led by Dr. Julian Gilmore, in which more than 1,000 or so patients were followed longitudinally for essentially definition of the natural progression of the disease. So these were patients that were untreated, predominantly wild type, about 70% wild type, the rest being hereditary. And in this analysis, you can see independent predictors of survival included age, NAC stage 2 and 3 disease. An increase in LVEF was protective. Of course, genotype was associated with worsening prognosis, specifically V122I, and a higher 6-minute walk test was predictive in this study. I'll next move into an interesting renal marker of disease. This is data also published by the NAC cohort, focusing specifically on albuminuria as a marker of CKD. And albuminuria was focused on rather than eGFR, given that it may change earlier than eGFR declines, and it's less affected by muscle mass, which is relevant in our amyloid patient population. And as you can see here, so included in this analysis were patients diagnosed between 2017 and 2023 who at baseline had a urine albumin to creatinine ratio measured. And clearly, the presence of macroalbuminuria, which was seen in about 5% of the cohort, portended a markedly worse survival compared to patients who had a normal urine to albumin creatinine ratio. And on multivariable modeling with adjustment for important comorbidities such as diabetes, hypertension, and disease severity, both micro and macroalbuminuria remain predictive of mortality. And so next, moving into an interesting neurobiomarker, that being neurofilament light chain, I wanted to highlight some of the data here. So what is neurofilament light chain? Essentially, it's a neuron-specific cytoskeletal protein that has been shown to be released in small quantities in the blood and also in the CSF with neuronal damage. And it's being increasingly studied as a biomarker, not just in hereditary TTR polyneuropathy, but also broadly in neurodegenerative conditions. In the middle panel is data from a sub-analysis of the Apollo study looking at patisiran versus placebo in hereditary polyneuropathy. And you can clearly see that with treatment over time, patients who were on patisiran had a decrease in their neurofilament light chain levels compared to those who were untreated who had an increase. This last panel is a more recent analysis done from the NAC cohort, looking at a longitudinal analysis of neurofilament light chain. On the top panel, you can see that neurofilament light chain levels clearly correspond to disease severity in hereditary polyneuropathy as measured by one's PND score. And in this longitudinal analysis, basically 4 groups were looked at. And I think most interesting is this sensory motor converter group. These were patients who at baseline had no neurologic symptoms, but over time developed both sensory and motor symptoms as measured by various scales and the presence of muscle weakness. And you can see in those patients, they had a higher level of neurofilament light chain at baseline and then a sharp rise over time, suggesting that this could be used as an early disease biomarker in patients with polyneuropathy. So next I highlight, I'm moving from individual prognostic factors to, commonly used staging systems that have been developed. Specifically, I highlight the NAC staging system published from the UK cohort from 800 or so patients that included both wild type and hereditary disease. And this prognostic staging system splits patients into 3 different groups based on NT-proBNP and eGFR. And clearly, if both markers were abnormal, your survival was worse. Median untreated survival was about 24 months, compared to if both biomarkers were above the threshold of normal, median survival was more than 5 years. More recently, the Columbia group looked at the importance of loop diuretic dosing and also the importance of loop diuretic dosing and NYHA class in terms of predicting survival in addition to NAC staging. And I think there were a few important messages from this analysis. One is the loop diuretic dose matters. So outpatient diuretic dosing at baseline was independently associated with survival when adjusting for various factors, including age, disease severity, proBNP, et cetera. And when loop diuretic dosing and NYHA class were compiled with NAC stage, ROC area under the curve increased from about 0.7 when using just NAC scoring to above 0.8 just highlighting the prognostic importance of these variables. So I want to next shift gears into talking about now disease progression, which is clearly an unmet need. Defining disease progression as an unmet need in TTR amyloidosis. You know, as more patients are living longer with this disease, and as we now may have not one but 3 therapies to choose from, defining disease progression is pivotal to optimizing outcomes. This unmet need was recognized by an expert consensus panel who published expert consensus recommendations for disease monitoring and progression in 2021, and this was based on essentially survey responses from those involved. And in this expert consensus panel, it was recommended to assess disease progression over 3 different domains: clinical and functional, laboratory biomarker, and imaging and ECG, with the thought that a patient must meet criteria from each individual domain to meet the definition for disease progression. And certainly that expert consensus panel is an important stepping stone as we think about refining the definition for disease progression. I highlight for our purposes a slide adopted from Dr. Matt Mara, which builds upon the domains mentioned, but also adds, importantly, patient-reported outcomes, which we'll talk about a little bit further. And I wanted to dive into some of the data for these individual metrics included in monitoring for patients with ATTR. So I'll start with the importance of 6-minute walk test in disease progression. We all know this test well. It's been utilized as an endpoint in recent AmyLite clinical trials, and it certainly is important in terms of progression. So shown on the leftmost panel is, this is data from the UK cohort, so patients were largely not on treatment, and the cutoffs used in the study were determined by ROC analysis, but you can see that at baseline, if patients had a 6-minute walk test distance less than 350 meters, this was certainly associated with poorer survival compared to above 350 meters. And then importantly, at 1 year, if there was a reduction of just 35 meters, that was associated with about a 1.8-fold higher risk of mortality. So moving next into changes in one's staging system, and this has been looked at as well in terms of if a patient moves from NAC stage 1 to stage 2, this has been shown to be associated with poorer outcomes as early as 6 months and out to 24 years. You can see associated hazard ratios there. And while I think this is important to recognize, it's a little hard to know how to incorporate this into clinical practice, as these are dichotomized variables, and the clinical significance may be a little murky from that standpoint. So I think while someone's increase in NAC stage or disease stage is important, I think probably more important is looking at the individual variables that compile these prognostic staging systems. And so going into that further, the individual variables I highlight from this well-done study looking at our NT-proBNP and diuretic intensification. So this cohort included patients from the UK, but also 7 additional centers that served for as external validation, with cutoffs again determined by ROC analysis. And you can see here that if patients had an increase in their NT-proBNP defined by greater than 700 and a greater than 30% increase at 1 year, as well as an escalation in their diuretic needs, that was associated with about a 3-fold higher risk of mortality compared to if patients did not meet those metrics. And this was also seen in the validation cohort, and a subset of those in the validation cohort were on treatment. So I think this brings up important questions that we can further get into in the discussion. Specifically, have NT-proBNP progression or ODI been recognized as clinical trial endpoints, and if not, should they be? With the notion being the data we show suggests that we could recognize clinical meaningful events earlier in patients with TTR cardiomyopathy. And so I'll last highlight patient-reported outcomes. Essentially, these are surveys that help assess health-related quality of life, symptom limitation, functional limitation in our patients living with this disease. A very commonly used PRO, as we all know, is KCCQ, or the Kansas City Cardiomyopathy Questionnaire, which certainly in non-amyloid heart failure has been shown to associate with mortality risk. And this was a sub-analysis done from the ATTR trial, showing that certainly patients on treatment had a higher odds of having improvement in these efficacy measures, KCCQ and patient well-being assessment. We don't have a lot of data on KCCQ progression in TTR amyloid in the real world, but I show here unpublished data from the Columbia cohort, in which KCCQ progression was defined as a greater than 5-point decrement over follow-up. And clearly this, on both univariable and multivariable analysis, was associated with mortality risk. And the last PRO I highlight is the COMPASS 31 score, which is an assessment of autonomic symptom limitation and was assessed in the neuropathy trials. So this is analysis from Apollo as well, showing that clearly with treatment, patients had an improvement in their COMPASS 31 score compared to untreated patients. And so while I presented data showing that we have come up with some answers and insight into disease progression, I think there are certainly still a lot of questions, starting with, if we come up with a set of metrics, what even is the optimal timing for monitoring, knowing very well that TTR cardiomyopathy is not a linear disease. There's a long period of patients being asymptomatic or having subclinical disease prior to symptom onset, and it's unlikely that those patients should be monitored at the same rigor and frequency as those who are symptomatic. Secondarily, what was alluded to earlier, what is the ultimate goal of defining disease progression? I think certainly we should move away from just trying to prevent patients from dying, but thinking more about preventing the morbidity of a heart failure hospitalization and improving overall quality of life. Very importantly, do our goals as clinicians align with the goals of our patients? What do our patients expect in this realm or want to look for in this realm? And lastly, what is the optimal intervention once identified? I don't think we have the data yet to say that switching therapies or combining therapies will help, but is that what needs to be attempted? Or is the only out right now to refer for advanced heart failure therapies, recognizing that many of our patients won't be eligible given elderly age? And so I'll end with a kind of a crude roadmap for designing disease progression, which our working group is focused on, starting with simply reporting on the current landscape for assessment of disease progression in TTR disease. Defining disease progression will certainly require, international multi-center collaborations, ideally for patients who are on standard of care therapy at this point. And consensus definition for these meaningful changes in parameters should ideally be, one, define what is clinically significant with validated data. And lastly, once we identify the best tools to assess disease progression, this should really be implemented with a patient-centric approach so we don't cause excess burden to our patients living with this disease. So in summary, the prognostic landscape of TTR, as we all know, is dramatically improving. Defining and identifying disease progression is pivotal to advancing the care of our patients further. And this approach to disease monitoring will certainly require a multi-parametric assessment. And I think the focus should be clinical relevance, reproducibility, and feasibility, for which larger scale multi-center studies will be needed, including patients on treatment. Thank you.
Thanks, that was great. I did want to see if any of the patients in the room or patient advocacy groups wanted to give their perspective a little bit, because I think this is very important to hear. If anyone's interested, please jump to the mic. There is an online comment from Dr. Merlini, who notes that the recent NT-proBNP grading response criteria probably should be more considered, because there's recent evidence that a complete cardiac response, that is return of NT-proBNP essentially to normal, is associated with survival that's similar to an age-matched population. So I'm curious if any of the people on the panel have any comments. Raj?
Yeah.
Yes, so recently there was a paper published on the graded cardiac organ response, so in which, similar to hematologic response, cardiac response can now also be graded into PR, VGPR, and cardiac CR, with PR being our traditional cardiac response definition of more than 30% reduction, it's 31 to 60%, VGPR being more than 60%, and CR being NT-proBNP of less than 350 nanogram per liter. So definitely, patients who have a cardiac CR, they have been shown to have similar overall survival as age-matched population without amyloidosis. So definitely, graded cardiac organ response, in my opinion, should be used going forward.
Great. Muriel?
Recently, we've come into a little quandary because of the great job that people are doing now with earlier diagnosis of TTR. We're getting phone calls from patients, and their concern is they were diagnosed with because of their carpal tunnel release, and they're sent to, and their orthopedic doctor is smart enough to send them to a cardiologist, and the cardiologist says, sorry, the PYP scan doesn't light up for you, so we'll just monitor you. So these patients call and they're in a panic because they've been diagnosed with a fatal disease, and they see from the data that the earlier they get on treatment, the better the outcomes, but they're told that they cannot get on treatment early. And of course, our AL patients, the minute their biopsy is positive, they're able to get on Darosib or D, but our TTR patients are kind of left in this little, limbo stage, and they're kind of panicky. So what can we do about that?
Anyone have any comments? I would submit that I completely understand where patients are coming from, because the first line in every amyloid article is a rare, life-threatening disease that, if untreated, but I'm not convinced that everyone who has carpal tunnel is going to develop cardiac involvement. I don't think there's very good data, but the best data I'm aware of is actually published by Finn Gustafsson from Finland, where he took a cohort of over 1,000 patients in Denmark who all had bilateral carpal tunnel releases. He didn't tissue type it, so we don't know that they had amyloid, but they were over 60, and he invited 125 men and 125 women about 7 years after their carpal tunnel release to come in for screening. And the positivity rate on a PYP was 4% in women and about 8% in men, and the highest group was men over the age of 70 who are not obese. It was 1 in 4, 1 in 5. So I think we may have activated a lot of people to be very concerned, and it's my clinical sense that some of these drugs are not very helpful for the orthopedic manifestations, though I don't think it's been well studied. And so I try to reassure the patient that right now that's the right approach, is that thankfully they don't have cardiac involvement, but they need to be surveyed for it. But I see, Julian, you raised your hand, so love your input.
Yeah, Julian Gilmore from the Amyloidosis Center. I just wanted to agree with Matt, essentially, that we know that ATTR amyloid deposits are incredibly common with advancing age, but we really don't understand that early natural history when patients may have, for example, ATTR amyloid deposits in the wrist and not have developed amyloid in the heart. And as you pointed out, Matt, we really don't know which patients will progress, which won't, and there's a lot of work to be done to understand what the risk factors are for patients progressing to clinically significant cardiomyopathy amongst that group.
Yeah. Ashu, I don't know if you had your hand still up. Want to make a comment?
No, I think Julian's made the comment which I was about to make. So I think we've encountered that with AL, where patients have a monoclonal gammopathy, where we don't understand the risk of patients developing AL. And hopefully we'll hear a talk later on, and we might come back to that discussion, but something like that needs to be identified for ATTR as well.
Sammy?
Thank you.
I would just say that in addition to a cardiac evaluation, if those patients are worried, is to have a neurologic evaluation. And if they don't have neurologic symptoms, which are, evidence of a peripheral neuropathy, then those patients should just be followed along.
Angela, running to the mic.
It's so Ashu sort of said what I was going to say, but in the plasma cell space, patients have a clone, they have a monoclonal protein, they live with it, they don't So as hematologists, we're more accustomed to sort of saying, this is a predisposition, just like hypertension is a predisposition for stroke, and so forth. So it's hard, but...
Yeah. I guess my personal struggle, I'm curious what anyone on the panel thinks, is there's almost so much to measure, and the timing of when we should be making these measurements is also unclear. So I don't know if anyone has any thoughts, how we go from where we are, where we have literally probably, 400 or 500 things that could serve as a biomarker for progression, and how do we whittle it down, both from obviously a rigorous data perspective, but also from a practical perspective, to not put too much burden on subjects. Anybody?
Can I make a comment?
I think this is a very fine balance, and we've spoken about this, how inherently patients who are sicker are going to get more frequent assessments, and that's probably okay. So, in my personal practice, I do a lot of these metrics simply before I see patients in clinic, and I'm seeing sicker patients more often. So I think that it has to be a balance, and I certainly don't think we should overburden patients who are doing well. We should really space out assessments and wait till we're approaching more symptomatic stages where we're thinking about different therapies.
Asha, what were you going to say?
I was just going to make a comment about biomarkers in AL. I think we need to have a correlation between the biomarkers and what actually happens structurally with amyloid deposits. We know from the work that Marianne has done with MRI scans that the NT-proBNP value in AL will decrease in 3 months, but the ECV will not have changed at that time. And then the NT-proBNP will decrease at a much slower rate, but the ECV will change at 9 months and 12 months. And the troponin value then drops off at 9 months and 12 months, and we are clearly getting different messages from different biomarkers. And one of the things we need to understand is what is the correlation between the structural change with the biomarkers. And then the other point in terms of the timing is the point that initially was raised in terms of the underlying clonal disease and the response to therapy. We are moving quite quickly in plasma cell dyscrasias in terms of achieving deeper responses more rapidly. And we are outgrowing the staging systems that we ourselves have developed because our responses are faster and deeper. So we need to have some sort of an adaptive design to figure out what our staging systems are with improving antiplasma cell dyscrasia therapies.
Yeah, agreed. Maybe one or two quick final questions. So Brian, you can go, and then Preston will be the last, if that's okay.
Great. Hi, I'm helping field questions from online, actually. So this kind of follows up on the question about carpal tunnel and surveillance for cardiac disease, but more about hereditary as well. So the question has to do with if someone has carpal tunnel, should they be considered for genotyping for a variant? And then also for people who have polyneuropathy, how does that affect surveillance for cardiac disease in the future if they're known to have variant neuropathy?
Yeah, I can tell you at our place, we do genotype patients who present with carpal tunnel, because the last thing I want to do is tell someone that I think we're very poor at encouraging cascade testing, and so that's what we do. I don't know what other sites do. And I think anyone with a neuropathy needs to be further investigated and may have indications for therapy, irrespective if they have cardiac involvement.
Yeah, I agree. We do a full cardiac assessment, including genotyping, and I've been surprised that there's been a hit rate in terms of carpal tunnel syndrome detecting subclinical cardiac disease and genotype positive patients.
Preston?
Yeah, Matt, it just occurs to me that as you talk about the potentially hundreds of biomarkers that may indicate that some people may progress, if there may not be one or two of these biomarkers, but basically constellations of them that may clarify what to do with these people that show up with carpal tunnel syndrome, and are they at risk for progressing or not? That could be a very interesting thing to look at those who do end up progressing, as far as what the constellations of these biomarkers were before they did start progressing. That's something the AI people are quite avid about doing.
Yeah, agreed. Well, I think this has been a great discussion. There'll be more. I'd like to turn the mic over to my colleague, Dr. Jan Griffin, who's at the Medical University of South Carolina, and she's going to present a proposal that the working group formulated with regards to the utility of cardiopulmonary exercise testing, which has been used in other diseases for amyloidosis in clinical trials.
Thank you, Matt. Good morning, everyone, and thank you for having me to present our proposal. So let me see if I can figure this out. I can. So this is the outline that we're going to run through. And I'm going to give you all just a little background on cardiopulmonary exercise testing, bearing in mind that not everyone in the audience is a cardiologist and is completely probably familiar with this test. So CPET, as it's known, is a diagnostic modality, and we use it to evaluate a patient's functional capacity. It provides assessment of the integrative exercise responses involving the pulmonary, cardiovascular, hematopoietic, and neuropsychological and skeletal muscle systems. And these are not generally adequately reflected through the measurement of individual organ system function. So exercise capacity as assessed by peak VO2 during a CPET is a dynamic objective variable, and it assesses cardiac reserve and peripheral adaptations to a reduced cardiac output. So peak exercise capacity, being measured in CPET, is the maximum ability of the cardiovascular system to deliver oxygen to exercising skeletal muscle and of the exercising muscle to extract oxygen from the blood. And here on the right, you see the setup for a CPET test. So we either use a treadmill or an upright cycle ergometer. There is an airflow transducer, which the patient breathes in and out of, and it measures the volume of inspired and expired air. And then a metabolic cart, which consists of a gas analyzer, a computer, and then we have a screen showing a 12-lead ECG and another screen that shows the physiologic changes as they're happening during exercise. Okay, now I'm stuck. Okay, maybe. Oh, there we go. Thank you. So what parameters do we measure? So the Fick equation that you see here states that oxygen uptake is equal to cardiac output, so your stroke volume by your heart rate, times the difference in arterial and venous oxygen concentration. And in a CPET, we measure it at peak exercise. So it's an objective assessment of the patient's functional capacity and an indirect assessment of cardiac reserve. So, for example, in patients with congestive heart failure, there's a marked reduction in stroke volume response to exercise. So as a result of this decrease in stroke volume with exercise, the VO2 is affected. And in other diseases, say of the lungs, skeletal muscle, hematologic system, it's generally the arterial or venous oxygen content that's affected. So the peak VO2, which is also the VO2 max, kind of the same thing, but VO2 max is more reflective of a healthy patient. It reflects the maximum ability of the person to take in, transport, and use oxygen, and it defines that person's functional aerobic capacity. Other parameters that we measure, I'll just briefly go through a couple of them. The respiratory exchange ratio, or RER, is a measure of effort of the patient. The anaerobic threshold is where we transition from aerobic to anaerobic metabolism in the exercising muscle. The VE-VCO2, which is a measure of ventilatory efficiency, or the ventilatory response to exercise, and the heart rate reserve, among others. So CPET has been studied over and over as a prognostic tool in advanced heart failure. Peak VO2 has been shown to be a good predictor of mortality, and a significant deterioration in maximal exercise performance frequently precedes a clinical decompensation. So CPET parameters can provide valuable prognostic information. So in a seminal paper by Donna Mancini and colleagues, which was published in 1991, they tested the hypothesis that peak VO2 could be used to identify ambulatory patients with heart failure in whom cardiac transplantation could be safely delayed. And they had three groups of patients. Group 1 were patients with a peak VO2 of less than 14, which they established as their cutoff. And if they had a peak VO2 of less than 14, they were accepted for heart transplant. Group 2 then were patients with a peak VO2 of greater than 14, and they were considered too well for transplantation, and their transplant was delayed, delayed. And then group 3 were patients who had a low peak VO2, so less than 14, but they were declined for heart transplantation for non-cardiac reasons. And what they found in the study was that patients who were in group 2, so peak VO2 greater than 14, so safe to delay, had a one-year survival, of 94% and a 2-year survival of 84%. The patients in group 3 who had the peak VO2 of less than 14 but were declined for transplant for other non-cardiac reasons had a 1-year survival of only 47%. And then the patients in group 1 who were accepted for transplant had a 1-year survival of 70%, probably an overestimation because those who underwent transplant were removed from the analysis. So their 1-year survival was likely not 70% with a peak VO2 of less than 14. It was probably closer to the group 3 patients. They then took it a step further and looked at intervals of peak VO2 and found that peak VO2 of less than 10 was a very, very poor prognostic indicator. And these patients had less than 40% 1-year survival. Patients with a peak VO2 of greater than 14 did pretty well, and there was really no significant difference above that. So the next parameter that has been shown to be a prognostic indicator in CPET is the VE-VCO2, which is a measure of ventilatory efficiency. And as your ventilatory efficiency deteriorates, your VE-VCO2 slope increases. So the higher the number, the worse your prognosis. So in this study from 2001, 123 patients with stable heart failure were studied, and all of these patients had a peak VO2 of greater than 18. So if you remember from the slide from Donna Mancini's paper, these patients should have a reasonably good prognosis. In this study, they dichotomized the VE-VCO2 slope at 34. And you can see here in the figure on the left of the screen, patients with a VE-VCO2 of less than 34, so, how do I go back? There we go. Thank you. The top slope here had a reasonably good survival of 93% at 3 years. But the patients with a VE-VCO2 of greater than 34 had a 3-year survival of only 57%. So a 4-fold increased risk of mortality at 3 years with the VE-VCO2 of greater than 34, despite a reasonably okay peak VO2. So what about CPET in ATTR? So Drs. Gilmore and Fontana and colleagues have analyzed their patient database from the National Amyloidosis Center, looking at CPET and the relationship to prognosis, but also CPET parameters in relation to amyloid burden on cardiac MRI. In this study, about 60% of the patients who underwent CPET were not on any TTR-targeted therapy, and about another 20% were within 6 weeks of starting TTR-targeted therapy or enrollment in a clinical trial. And what they found was the mean peak VO2 was significantly decreased across the board. They had patients with ATTR-CM, ATTR mixed, and ATTR polyneuropathy. And you'll see here on the right-hand side of the screen, the peak VO2, percent predicted peak VO2, and the peak workload were significantly decreased in ATTR-CM and ATTR mixed compared with ATTR polyneuropathy. And that was seen visually here on the left. You can see the peak VO2 here for the ATTR mixed and CM significantly lower than the polyneuropathy group. They also then looked at the VE-VCO2 slope over here, and they found that in the cardiomyopathy group, a VE-VCO2 of 38 compared to lower 34 and about 29 in the other two groups, again, significantly higher and indicative of a poor prognosis in this group, visualized here again on the left of the screen. So they also looked at CPET parameters according to amyloid burden, and this was based on extracellular volume measurement on cardiac MRI. And in these patients, there were over 300 patients that they were able to evaluate. And the percent predicted peak VO2 was reduced in all of these patients. But the degree of reduction of peak VO2 was progressively more severe with increasing cardiac amyloid infiltration. Here on the bottom, you have the ECV on the x-axis and the percent predicted peak VO2 on the y-axis. And you can see that for every increase in the extracellular volume, the peak VO2 continues to decrease. They find the same for peak O2 pulse at levels of amyloid infiltration that were no amyloid infiltration or mild to moderate infiltration. You know, the 6% predicted peak O2 pulse was reasonably okay, but as you had severe amyloid infiltration, this was also decreased. And again, for VE/VCO2 slope, only the patients who had no amyloid infiltration on their cardiac MRI had a reasonably good VE/VCO2 slope. But for any increase in amyloid infiltration on MRI, the VE/VCO2 slope continued to increase. And you can see the correlation here on these plots for those variables. So in the study, they had a mean follow-up of 24 months. Impairment of functional capacity they found was highly prevalent in all patients via various mechanisms. They found that the peak VO2 and peak systolic blood pressure were independently associated with patient survival in this ATTR group, and that's shown here in the Kaplan-Meier curves on the right-hand side. The top for peak VO2, as the peak VO2 decreases, the survival decreases, and then on the bottom, as the peak systolic blood pressure decreases, the survival decreases. And then finally, CPET has been used in clinical trials before for obstructive hypertrophic cardiomyopathy. The Explorer HCM study, which was an evaluation of the efficacy and safety of mavacamten, which is a cardiac myosin inhibitor, in symptomatic obstructive hypertrophic cardiomyopathy. The primary endpoint for this study on CPET was a 1.5 mil per kilo per minute or greater increase in peak oxygen consumption, peak VO2, with at least one NYHA class reduction, or a 3 mil per kilo per minute or greater increase without any worsening in NYHA class. And the CPET was performed at baseline and at week 30. And what they found was that about 37% of the patients in the mavacamten group reached the primary endpoint, and versus 17% in the placebo group. So almost a 20-point absolute difference between the two groups. And then based on this study, the FDA approved the use of mavacamten after this 30-week trial. So now to our study, or our proposal. So what we propose to perform is a multi-center retrospective study of patients with ATTR cardiomyopathy and AL cardiomyopathy as well at some point along the study, who are on cardiac amyloidosis targeted therapy. And that will be most of our patients. We want to identify those who have performed a baseline CPET prior to or after initiation of the therapy, determine the association between baseline CPET parameters with outcomes, those being mortality, transplant, or LVAD, even though none of our amyloidosis patients should be getting LVAD, but we all know some of them do, and heart failure hospitalizations. And these, again, in patients on disease-modifying therapies. We want to determine if baseline CPET parameters correlate with invasive hemodynamics in patients who have paired studies. And then in a subgroup who've completed or agree to complete a repeat CPET, we want to evaluate change in CPET parameters over time in those on TTR-targeted therapy, so whether they improve, stable, or worsen. Why do we need this study? Well, as I've shown you, cardiopulmonary exercise testing is prognostic in end-stage heart failure, and we use it for all of our patients who we're evaluating for heart transplant or LVAD in that area of cardiology. As a surrogate, change in 6-minute walk test has been used in many ongoing and previous clinical trials for ATTR. And as Dr. Vaishnav showed you, greater than 35-meter reduction or 5% relative reduction associated with an almost 2-fold mortality at 1 year. However, I would argue, and we would argue, that CPET is a more objective assessment of cardiopulmonary status, and it provides a lot more information on the functional status of the patient. CPET-derived parameters have been shown to be strong predictors of mortality in many other studies, and can we use CPET to assess prognosis, to evaluate progression of disease, and maybe patients who are responding or not responding to therapies for ATTR cardiomyopathy at an earlier stage in the disease process. So how will it benefit patients? Well, it will hopefully provide additive objective assessment of prognosis at baseline. So those patients who come in and ask you, what's the story? How long am I going to live? How sick am I? Maybe a CPET is the way to help them with that, with some of that information. Assess response to therapy, again, maybe at an earlier time point than we've seen historically in clinical trials, particularly in the more current era with all of the medications and therapies that we have. And assist in decision-making of when to change therapies in non-responders or in those progressing despite being on TTR-targeted therapy. Our expected regulatory need, hopefully provide a robust and reproducible endpoint for clinical trials. provide multiple prognostic parameters. As I've shown you, there's many, many variables that we can get from a CPET, which can be objectively measured, on which basis to approve new drug therapies, and maybe, again, an earlier indication of response to therapies, and could potentially shorten the ever-expanding time frame and huge cost which is required to run a clinical trial. So our objectives, firstly, to determine the association between peak VO2, VEV-CO2, which is the ventilatory efficiency, heart rate variability, circulatory power, and any other parameters which the group thinks is reasonable, with cardiac outcomes at 1 year in patients on TTR-targeted therapy. To determine if there is independent prognostic value of CPET variables in addition to the known baseline risk factors such as disease stage as determined by the scores that we use regularly, the NAC score, the Columbia score, and any other prognostic indicators that we have. To evaluate the correlation between these identified prognostic factors that we deem from our CPET analysis with invasive hemodynamics. So do the CPET parameters correlate with the invasive hemodynamics in these amyloid patients? And to evaluate then finally the change of CPET variables in those who have paired CPET pre and post initiation of targeted therapy, and in paired CPET in those who have been on continuous therapy for the entire period. And then we want to compare these against other more subjective patient-reported outcomes such as KCCQ, and also hard outcomes like death and transplant. So this is our target study, as I've kind of already gone over, patients with ATTR and on therapy. These are the data that we'll require, which I'm not going to go through, but up for discussion. We have some potential collaborators already identified, but we will take anyone who's willing to help. So Columbia, Johns Hopkins, and ourselves at the Medical University of South Carolina so far. We've created a data sheet and have discussed potential for involvement of federated analytics for data analysis, but that is yet to be, that's for further discussion and yet to be determined. So the timeline for the first 3 objectives, probably about 6 months to get the data and analyze it with the goal for manuscript within the first year from initiation of data collection. And objective 4, where we need to repeat the CPET in patients to assess for change in CPET parameters, would be about another 6 months from baseline. I'm not going to go through the challenges and barriers, but there are many. Data sharing, small population, reproducibility of CPET at different centers are all relevant, and also patients on lots of background heart failure medical therapy, which is not standard across all institutions or patients. And now to the key questions for feedback. So there are many. So academic scientific questions. Is this a useful endeavor? Is there any point in doing this? Is there a role for the use of CPET in clinical trials for ATTR cardiomyopathy? Will it provide us any benefit over the standard 6-minute walk test? I'm biased, I think it will, but that's up for discussion. And do you think that we could improve CPET variables with any disease-modifying therapies? Or would it help us to see if we are slowing or preventing a decline in disease progression? For industry, is this cost-effective? 6-minute walk test is very cheap. CPET, not so much. It's not an expensive test, but it's not as cheap as a 6-minute walk test, making someone walk up and down a hole. Would the use of CPET as a primary endpoint limit site identification for inclusion in clinical trials? You know, they're done at all centers. I can tell you that do transplant and LVAD are going to have a CPET program, but not every center in the country has a CPET program. And what if we demonstrate that some therapy will prevent declines in CPET variables but don't affect any improvement? Is this going to affect the desire to use this in a clinical trial? And then for our regulatory colleagues, can we inform the use of CPET as an endpoint or as a surrogate endpoint in clinical development and regulatory evaluation, particularly for early phase clinical trials, phase 1, phase 2? Could data from this study address any concern the regulatory authorities may have regarding consistency and reliability of CPET across medical centers? Again, as I've mentioned, with respect to all the other medical therapies that they may be on. And what is this magnitude of variation? And if CPET is prognostic and associated with their clinical measures, could it be suitable for supporting accelerated approval in ATTR cardiomyopathy? So again, shortening clinical trials and making them maybe a little bit more cost-effective. And those are now up for discussion. Thank you.
Fantastic, thanks. We have about, I think, 10 or 15 minutes for discussion. I see, Justin, you have your hand up online, so I'll jump to you.
Yes, thanks, Matt. And Jan, that was a wonderful presentation. So my question to you and the panel, obviously, I think it's, CPET gives us a wealth of information that's both physiologic and prognostic. But I think the one key question is I think hypertrophic cardiomyopathy with obstruction is a very unique disease that was clearly responsive to a targeted therapy on a very specific physiologic hemodynamic mechanism. But when we look at heart failure clinical trials, it turns out that CPET or peak VO2 improvement is quite a high bar. And, for most pharmacotherapies, where I'm not aware of many, maybe you are, but I think very few, if any, therapies that have demonstrated an improvement over time. So, what we do know is that exercise in particular can improve CPET parameters and high intensity interval exercise. But so I think the question for the group is, yes, this is a wonderful project, it's a brilliant idea, but even if we see that these parameters are prognostic, do you envision that they will be modifiable with therapy?
Yeah, I think I'll give my opinion, I think it's a great question. I was going to ask my industry colleagues, it could be that a therapy would prevent the decline that we'd see in the placebo group. I'm not sure, to your point, given the complexity of what CPET is measuring, the age of the population, and the multiple mechanisms that could limit functional capacity, that we wouldn't show a benefit. And I don't know how, to be honest, such a compound would ultimately be marketed if it slowed the decline rather than improve things. So I think your questions, in my perspective, are very good ones. And I'd be curious to hear, I see Norm's at the mic, just if there are lessons learned from HCM, as Justin's pointing out. Very different physiologies, but my sense is CPET has been accepted by regulators for years as an endpoint that's registerable. It's not something you need to reinvent the wheel. I think we as a community need to make sure we can make the measurement in these individuals and make it, in a consistent and manner over time. But any thoughts you may have?
Yeah, I did want to say pretty much what you just said, which is that CPET is a clinical outcome. It's a measure of your ability to do work. And so accepting it is not a problem. Conceivably, showing less of a decline is acceptable, but it'd certainly be acceptable to us. May not be acceptable to your patient community.
Right.
And as far as this assessment of existing data is concerned, I thought it was useful to point out that for this to be useful, it would need to manifest an effect from an intervention fairly early, right? Because otherwise you might as well be looking at the hard outcomes. But it's also true that once you start having missing data because of hard outcomes, you can't interpret the exercise data at all anyway.
Yeah, I think your point is well taken. There's a much higher morbidity and mortality in TTR amyloid than there is in HCM, and so that would be a big confounder. Donna?
Yeah, just a quick question, an interesting project. In the data that's currently available, have you evaluated the correlation of the different CPET parameters with 6-minute walk?
Not that I have seen in the literature, but maybe someone else knows. I haven't seen them compared.
Because it seems like that publication from the UK has a fair amount of patients. Did they evaluate 6-minute walk?
I don't believe so. I think Dr. Gilmore is online, so I'm not sure if he could answer that question.
I see Mariana has her hand up, so I don't know if it's in response to that question or any insight she could provide or a different question, but take it away, Mariana.
Thank you. Yeah, it was for a different question, but no, we didn't assess the correlation between the CPET and the 6-minute walking test, but it's certainly something that can be done if we believe it's a meaningful correlation. But in a sense, so we didn't do it not because we didn't have the data, but just because I wasn't entirely sure of what was the intention behind trying to correlate the things, because they assess quite different things. There would be a correlation between the CPET parameters and the 6-minute walking test, but then how to assess what would have been expected, how good is that, and everything. That's the reasons why we didn't go there, just because I didn't know what I was trying to answer when we were going to do that analysis. And another point that I wanted to make is that one of the things that we need to bear in mind is that we need to make sure we don't take the meaning of certain parameters of CPET in heart failure, translate them to amyloid. Because, for example, in heart failure, the VV-CO2 slope is an independent predictor of mortality together with the VVO2, and that's the peak VO2. And that's a very big, in the sense, statement that has gone through the literature all the time and is clearly there. When we analyzed the 500 patients with the ATTR amyloidosis, interestingly, VV-CO2 wasn't predicted. And the correlation with several aspects of disease parameter was very different from what you would expect. And we believe that that is mainly driven by other things like direct amyloid infiltration in the lung, which of course are not present in heart failure. So the ventilatory efficiency, so the slope in amyloid, it actually might represent something different from what it represents in heart failure. So I think there is a bit more work to do on understanding the different parameters, the meaning in amyloid, which I think can be quite different. And one beautiful example is the slope. Is not the same, or it can't be used the same way we used in heart failure. It can't be used to independently predict prognosis. It can't be used to track disease exactly in the same way. So I think there is a bit more understanding about the CPET in this disease that has to be done.
So Marianne, just to follow up, if I'm understanding you, you're saying that peak VO2 is predictive of survival, but VEV-CO2 is not. Is that?
So yeah, VEV-CO2 was predicted at univariable analysis, but wasn't independent. Whilst we know that in heart failure, they are independently predictive of prognosis. Actually, we use one and the other one to classify patients. And instead here was extremely clear that VEV-CO2 didn't have the same role of independently predictive as in heart failure. And there was also a bit of dissociation between, for example, the disease severity and the slope. And our interpretation was, I think, because we think there is something independently that goes on in the lung, it's not just like it is in heart failure, activation of the chemoreflex response, activation of the neurohormonal system that leads to hyperventilation per production of CO2. So, and is entirely conceivable because we know from autopsy studies that the alveolar capillary membrane is heavily infiltrated there. So, for example, what is the role of the lungs in the exercise capacity in these patients? Is diffusion impaired by the direct amyloid infiltration? So I think there is a lot to explore on the differences of CPET in amyloid versus CPET in heart failure.
John Vest.
Yeah, hi, John Vest from Alnylam. In the slides and in the commentary, a lot of comparison to 6-minute walk test, which you in the slides I think commented that it was inexpensive. I will say that in a large clinical trial, while this is a 6-minute walk is very, very complicated to administer and do well in the trial. It sounds simple, but it really leads to a lot of headaches. I'm just curious, but it can be done anywhere, and there's a lot about this that is really attractive. There's a more objective sense to it. It provides a lot more information, but I am curious to hear your thoughts on its applicability in a large, global, multi-center trial. How complicated is this to do? Because that issue around identifying sites and those types of limitations are, they're very real if we're thinking about large-scale clinical trials.
Yes, I don't disagree with you at all on that point. I think from your first question as to how how applicable would it be to carry it out in different sites across the world. I guess my point showing the hypertrophic cardiomyopathy or the Explorer study was that was a multi-center international study, and they were able to perform it in that study. So I think that I guess my only concern is that it might limit site identification in that, across the broader spectrum, not everywhere has a CPET machine, but it is, and given that it's an objective study, you can specify what protocol you want to use, you can specify the machines you want to use, and you have, an outline for the staff performing it. I don't foresee it being very difficult to standardize across institutions. I think, though, that like anything else, that you would need a core lab to read them. It couldn't be read locally because then you're going to get a lot of variability between centers. But I think the same way as we perform any test, having a core lab, having an outline or a protocol of how they're performed and what exercise test you're going to use on the treadmill or on the ergometer or whatever you're using, I think with that we can potentially standardize it. I won't say easily, but it is standardizable across centers.
Okay. Vishali?
Right, so as a non-cardiologist in the group, so at Boston University, Boston Medical Center, we have two different CPETs. One is on the treadmill, one is on a bike. And we also have invasive CPET. So are you able to pull data together from different types of CPET? Moreover, these patients have other comorbidities. And when ATTR amyloidosis patients are put on a treadmill with lumbar spinal stenosis or some other, osteoarthritis, forget about even lumbar spinal stenosis, osteoarthritis, they can't really perform treadmill appropriately. So, that is how we are doing the bikes, which are stationary bikes. Also, autonomic neuropathy patients, they can develop significant hypotension during all these exercise studies. So I feel that we choose and select patients who undergo CPET just because, we are so afraid that they're going to have an event while doing it. So, that would be my only, and the data transfer agreements between institutions is a cumbersome process.
Yes, it is.
As we all have known. And it can take anywhere from 6 months to a few years. So, I think that federated analytics for any of these projects where you have to combine data, I think sounds more and more attractive.
Yeah, we'll hear, I think, more this afternoon, hopefully, about the benefits of federated analytics. But I think we have time for two more questions. So Spencer and then Ricardo.
Thanks, Matt. Thank you for the wonderful presentation. So I think, first of all, just the first question, is this worthwhile? I think this is a worthwhile project. I try to write down some of your questions here. But along with John, I think the question on cost effectiveness, which you asked for industry, is it's kind of a loaded question because if you can see change in CPET faster, then it's certainly not more expensive. Right? So if you can do a shorter trial than you can with 6-minute walk test, it's more cost effective, as well as if you see, an earlier signal, but also just a better signal. I think as we're talking about survival trials are taking longer, so our 6-minute walk test endpoints are taking longer to reach too. So can CPET get there faster? Another question, just on the proposal, in terms of patients. Is this the idea to enroll kind of all comers at different stages? Just kind of thinking about that, if you're doing this with current sort of standard of care, you would expect, some stabilization or potentially decline. And just curious if you think maybe if you studied in later stage patients, if you could get to an answer quicker, at least on the decline portion of that. I do think obviously there's hope for the future that you can have therapies or combinations of therapies where you do see improvement in CPET.
That last point is a very good point and not something we actually really discussed as to what stage of patients we would think about including in it. I think it's a really good point that if we have them too early, that we're not going to potentially see any change when we put them on therapy. I think the whole point, I guess, of this proposal is more so to see what we find and to see if we can see a change, if it might be useful, rather than because I don't know. I don't know if CPET is going to help us see an earlier change in people on therapy. But if we can do this study and we determine that, yes, we can see an earlier change, then it's going to be useful for clinical trials. But that's kind of the first hurdle, is to see what we find, essentially.
Okay, Ricardo?
Yeah, this is...
Ricardo Rocha from... Oh, go ahead.
Mariana?
No, I just wanted to add that I didn't say that before, but I think this is an amazing proposal and was an amazing presentation. So thank you so much for bringing up this topic of the CPET so clearly and so well with an amazing project. So I just didn't say that, wanted to say that.
I would agree with that, Mariana. And I actually, I'm Ricardo Rocha from Intellia Therapeutics, and what I wanted to say is, very quickly, we get into a debate of, is this particular way to assess patient status or the patient prognosis the right way to do it? And it probably is not going to be either one in isolation. And I think the point was brought up about that patient that is diagnosed and comes to the doctor and says, what do I do now? And I think what's going to be really important from the overall work of this group is, how do we bring all of these multiple markers that seem to be related to progression, how do we use them, perhaps not in isolation, but in aggregate, to, number one, answer some of the academic questions, and you did a really nice job breaking down the academic versus industry versus regulatory questions, but really, what's the definition of success, particularly with the patient in mind? Is it keeping a patient from dying, or is it keeping a patient stable? Or somebody brought up before, is there a way to determine treatment response or treatment improvement? And I think all of these is work that is starting now, but I think we need to keep the patient in mind and realize that for us as an industry, what's going to be really important is, how do we use all of these to be able to not just demonstrate morbidity and mortality, hard endpoint benefits, but how do we do trials earlier in the disease, and perhaps in the future, can we actually prevent onset of the disease, or at least, and ideally, regression for those patients that are already sick?
Yeah, all great comments. In the last presentation, I'd like to invite Jing Zhang from Protego up, who's going to talk about the AmyLite assay for prognostication in patients with AL amyloidosis.
Good morning. It's my privilege to present our plan to evaluate AmyLite assay, which is a new assay that detects a de novo biomarker called DLCCD in blood samples as a quantitative prognostic biomarker in AL amyloidosis. So the result of the study will be submitted for publication, but more importantly, we hope the results of the study will help address unmet needs in evaluating a new assay that could be served as a fast and accurate diagnostic and prognostic assessment using baseline samples, and a way to follow treatment response as well as disease relapse. And on the regulatory end for drug development, we hope this study will generate a large data set that evaluate the potential of this assay to serve as a quantitative AL amyloidosis specific progression biomarker that can monitor response to therapies. And here we're talking about therapeutics agnostic, and also even as an endpoint or a component of an endpoint that reasonably predicts outcome, enabling accelerated regulatory approval. So in the next few slides, I'm going to tell you why we think AmyLite assay and DLCCD biomarker has such potential. We know that in normal plasma cells, they make immunoglobulins. They also make up to 26 milligrams per liter of polyclonal lambda free light chain. And in contrast, in AL, the clonal plasma cells make on average 20 times more of the free light chain protein. And in majority of those cases, it's a lambda free light chain clone. And in AL, because those free light chains are unstable, when they're secreted into circulation, they form a wide range of protein structures, and sometimes they get fragmented. A lot of those constructs are highly toxic to tissues and organs such as the heart and the kidney. And because every AL patient has a unique free light chain sequence, and that generates a diverse spectrum of instability and amyloidogenicity that cannot be captured with the current free light measurement of total free light chain levels. So based on this AL amyloidosis disease mechanism, we hypothesized that unstable or amyloidogenic free light chain, even at levels close to or below the normal range, may still drive disease pathology. And if we can develop a method that can detect and quantify unstable free light chain, then we should have a very powerful tool for AL diagnosis and prognosis. And that is the idea behind the AmyLite assay. So here is a diagram showing how this assay works. So AmyLite assay starts with biofluids such as serum or plasma, which contains lambda free light chain. And then lambda free light chain is a homodimeric protein, so that means two identical protein chains are within the same molecule. In this case, it's linked at the C terminus by a disulfide bond. And the top part of this molecule is the variable domain. And as the name suggests, it varies in every patient. And the bottom part is the constant domain. So AmyLite comprises of two steps. The first step is a limited proteolysis step that takes advantage of the difference in protein stability between amyloidogenic free light chain and the normal stable light chain. And then we were able to find the optimized condition and also using a specific protease that can cut in between the variable domain and constant domain. And then the more unstable the lambda free light chain is, the more likely it will get cut. And then this gentle cut generates this dimeric light chain constant domain, or DLCCD biomarker. And then as the second step of the AmyLite assay, we have developed a highly sensitive immunoassay to quantify the resulting DLCCD biomarker. And this entire assay can be done within a few hours. And so next we looked at the specificity of the AmyLite assay by testing a number of plasma samples from healthy volunteers, multiple myeloma patients, lambda AL patients, and ATTR patients. And we subjected the plasma samples to our limited proteolysis and then used the immunoassay to quantify the resulting DLCCD biomarker. And you can see on this left side graph, and on the log scale, DLCCD biomarker is prominently elevated in the lambda AL patient sample, but not so much in the other cohorts. And we also noticed that there is little correlation between the DLCCD biomarker and the free light chain level in each of these plasma samples that indicates that DLCCD is an independent biomarker. And this AL patient samples are pretreatment samples or baseline samples that from 50 confirmed lambda AL patients provided by Dr. Mutka and Dr. Dispensary from the Mayo Clinic. So in order to understand what the clinical relevance of this DLCCD biomarker, Dr. Mutka did analysis looking at the patient's overall survival that's shown on the right-hand side. And the patients, lambda AL patients were separated into two groups based on their baseline DLCCD level, with 25 patients in each group. So as you can see here, the patients with lower baseline DLCCD level in blue, and they had median survival of close to 10 years, while the patients with higher baseline DLCCD level in red, they had median survival of just over 1 year. So, and the difference is quite significant. So this encouraging preliminary study shows us that the DLCCD biomarker may really be detecting something that's toxic and pathologically relevant in the patient samples. And we know that in AL cardiac amyloidosis, the cardiomyocyte insults cause organ damage, and then progressive organ damage would then lead to cardiomyopathy phenotypes as well as upregulation of downstream signaling pathway, including elevation of the biomarkers such as NT-proBNP. However, those markers are influenced by many other factors, such as genetic background, comorbidity, medication, stress, diet, etc. And then in contrast, the toxic free light chain, now detected by the DLCCD biomarker, causes direct insult to the cardiomyocytes and other tissues. Therefore, we think DLCCD biomarker is uniquely positioned as a quantitative prognostic biomarker for lambda AL. And so this research project intends to evaluate AmyLite's potential for AL diagnosis and prognosis. So here are the three research objectives. Number one is to verify the prognostic utility of AmyLite. We want to confirm if DLCCD biomarker can indeed predict overall survival using the baseline samples independent of treatment. This will help us establish the link between unstable free light chain and AL disease pathology. We would also like to understand if the prognostic utility is based on individual patient level or more based on a cutoff value. And the second objective is to probe DLCCD utility in predicting treatment response, i.e. whether DLCCD level, especially after clonal plasma targeted therapies, correlate to the lack of organ response or mortality in some of the patients. The third objective is to delineate the disease driver. I think we had some discussions around that before. Is the disease really caused by clonal plasma cell proliferation or free light chain instability or excessive organ damage? So this will help us better understand the disease mechanisms and come up with better therapeutics or combination of therapeutics for AL treatment. And in order to accomplish those research objectives, we will need access to longitudinal retrospective patient samples, serum or plasma, as well as corresponding de-identified clinical data. So we intend to separate the patients into pre- and post-DARA era and also patients with varying treatment outcomes. I won't go into the detail on this list. And this is a proposal of how and who and how we're going to do it. As you can see, it's a large undertaking that it's a collaborative effort between clinicians, drug developers, ARC, and the FDA. So Protego will run the AmyLite assay on samples that mask those identities, and then eventually will be unmasked by a third party. And we're still working on the logistics of this project as well as funding support. So if any entity is interested in learning more about this project, please reach out to Kristen or me. And here are some key questions. We'd love to have feedback from the audience and the regulatory agencies here. First of all is what are the requirements to establish an AL prognostic biomarker from a regulatory standpoint, both for treatment decision as well as to guide drug development? And if we get such a prognostic marker, how to best use this marker in those contexts? And we'd also like to gauge the interest from the community because this is a large undertaking. We won't be able to do it at this speed and the scale without such support. And then also very importantly, I think it has been touched upon a few times today, what other clinical parameters can be combined with this AmyLite DLCCD biomarker for better prognostic prediction. And with that, I thank you for your attention. I think we'll move on to the discussion. Thank you.
so anyone's welcome to comment. I'll start. I guess I was a little intrigued. I know there are a lot of patients, I think, Ray, you've done a lot of this work, who have, myeloma who are misdiagnosed, and I saw in your graphs that there were a few people in the multiple myeloma group that seem to have toxic free light chains, and I wonder, scientifically, if this could be utilized as well to just identify a cohort who, God forbid, is missed by clinicians. Any thoughts?
So the multiple myeloma samples, we haven't had a whole lot of them. And some of those, we know the ones that have upregulated signal are true lambda light chain multiple myeloma patients. And this could be, as you alluded to, could be some of them have higher levels of unstable free light chain. It could also be because we know that in multiple myeloma, the free light chain level could be 100 or even 1,000 times higher. It may be an assay issue, so we need to know the free light chain level in those samples, and then maybe eventually we'll have to do a ratio on that. So those are all possibilities.
Yeah, thank you very much. Two questions. One is, did Dr. Mukhtar do a multivariable analysis? Because all of that data is annotated, and the question that arises is whether or not the level of the free light chain or the level of what you're calling the unstable light chain, makes it through a multivariate analysis as significant. Did he do that?
Yeah, I don't know if Dr. Mukhtar is online. Maybe
seems like Dr. Dispensary is coming to answer.
Okay, thank you.
It's a small sample size, so hard to do multivariate, but it does drive it out. It is significant and independent of the light chain levels.
Okay.
So, yeah. And the other thing, I think what you were asking, is this assay potentially useful for diagnosing AL in unsuspected people, be it myeloma or MUGUS? And the answer we would expect and hope is yes. So multiple utility.
Yeah. Vishali?
But just to kind of clarify on that, most of the myeloma patients and most of the MGUS patients are kappa, and this assay is only for lambda. I know that Shin is working on the kappa, and hopefully we will have it. And the follow-up question was about the ATTR samples, because we know that, about quarter of patients, or even in our series, 39% of patients have monoclonal gammopathy associated with ATTR wild type. And did you have any of those patients in there?
Yeah, so that will be in this proposed project. We will test some of the MGUS patients and the TTR patients.
And then obviously whether this assay also helps us to differentiate between organ responders and non-responders.
Correct. Thank you.
Ashu, you want to make a comment?
I think Vaishali and others have made some of the comments, but I think there are two quite different uses of this assay. One would be at baseline, but one would also be after treatment. Because at the moment, we'd still be having plasma cell-directed therapies for all the patients irrespective. And the low-level persistent light chain after treatment is where this assay would be really critically important to decide whether these patients would benefit from further therapies, particularly if they have got very unstable free light chains. And then the TTR point that Vaishali raised is important because it may become a very helpful assay in ruling out AL in patients who have a positive PYP or a DPD scan where you have abnormal light chains. If you can show that this assay allows us to rule out AL if this assay is normal, it might save the patient a lot of investigations.
Yeah, great points. Eugene?
Eugene Berlowski from Philadelphia. My question was more regarding MGUS, what was brought up earlier. We often see these patients with MGUS or smoldering myeloma with HFpEF. How many of those actually have monoclonal gammopathy of cardiac significance, so to speak? And what markers can we use to identify those patients? Do we biopsy all of them, do MRI, or can AmyLite help with that?
Yeah, that would be the hope. We really hope to be able to access a lot of those samples, especially longitudinal, even before they develop clinical symptoms, and follow them over time and see when we can see that level.
Yeah, I would submit we've tried, I think, for years as a community to try to develop a broad biobank if we could. I mean, if you think sharing data is hard, imagine sharing samples, but projects like this remind me of the project you assign to the fellow and then you forget to tell them that they have to, like, you know, come up with 3 more variables and they have to go through another 1,000 charts over. And so I wonder, as a community, maybe we can leverage this project, but think through a way to collect samples that I think people are using or collecting at all sites and figure out some way to develop an international repository and biobank so that additional questions that come up could be addressed, because I think it's really sorely needed in the field.
So I don't know if like existing trials have SERA saved, but that would be an ideal, you know, trials that have been completed, whether that could be used there because you have well annotated samples and, you know, perhaps even baseline would be of interest, but even, you know, 1 year or whatever. So that would be another really great opportunity for collaboration.
Rick? Yeah, could just move.
I'll just suggest. Yeah. No, I think the AML is a question of the organ group that to me is really interesting. So I make sure in your cohort that you have people with renal disease, cardiac disease, and then even isolated one or the other, so you can have disentangle not prognosis, but organ response based upon those groups.
Yeah, that's a great point. We will be collecting those organ status as well as part of the clinical data set.
Okay, if there are no other questions. Oh, Ashu, you have, sorry, I think your hand's back up.
One last point as a suggestion to make things easier. Given the complexity of sample handling and data sharing, if the assay could be set up in individual laboratories where we've got big sample repositories, like we probably got samples from maybe 1,500 patients that we could easily run if the assay was set up. And I'm sure Vishali and the Mayo Clinic and yourself, Matt, will have sample repositories where the samples could be run. So it would be another way of fast tracking the project, at least from a prognostic perspective.
Yeah, that's a great suggestion, which is, you know, send the samples to a single repository and get the answer and combine it with the site-specific data right away. All right. Well, I thank you all for a very robust discussion. I think it's time for lunch. So we'll convene again, I think, at about 1 o'clock, 1:10.